Okay, good morning, everyone. I guess we can get started. Welcome, everyone, to our fifth annual University of Miami Neuroengineering Symposium. My name is Dalton Dietrich. I'm the Scientific Director of the Miami Project to Cure Paralysis and co-director of the Neuroengineering Institute with my colleague, Dr. Sarud Rajguru in the Department of Biomedical Engineering. A special hello to our colleagues throughout the state of Florida that have contributed to this program this year, as well as other colleagues throughout the United States. The Neuroengineering Institute was formed at the University of Miami around 2017 to initiate new collaborations with neuroscientists and engineering community at the University of Miami. And we formed that with Dr. Oscar Azamar back in 2017. Now, before that time, we did have some interactions between engineering and neurosciences, but the Institute was brought together specifically to bring more engineers over to the medical campus, and also to bring the next generation of students and trainees in this very exciting area of research. So we're really excited to announce that we'll be starting a new combined BS and MS program in neuroengineering next year. And this is going to provide our students with the special skills to solve complex problems in that interface between neuroengineering, medicine, and neuroscience. Now, these types of multidisciplinary programs really would not be possible across the three campuses, across different schools, without the leadership at the University of Miami. We really want to thank our medical dean, Dr. Henri Ford, and our new engineering dean, Dr. Biswas, who have actually put together a vision for the future of neuroengineering at the University of Miami. So it's my pleasure right now to introduce the Dean of Medicine, my boss, Dr. Henri Ford, with some introductory comments. Henri, please.
Good morning, everyone, and welcome to the 5th Annual Neuroengineering Symposium. As Dean and Chief Academic Officer of the Miller School of Medicine, I'm particularly excited about the symposium that is bringing together investigators from different areas of expertise to tackle challenging research and clinical problems. I'm particularly happy to report that this year, we have researchers from several universities joining us, including Florida State University, the University of Florida, the University of South Florida, Kentucky College of Engineering, and Washington State University. I bid all of you a heartfelt welcome to the University of Miami and the Miller School of Medicine. This symposium is the result of growing interest in interdisciplinary programs established with the colleges of engineering and arts and sciences to promote collaborations between the neuroscience and engineering communities for the advancement of new technologies and to foster synergistic interactions between our research and clinical programs. The theme of this year's virtual meeting is Neurotechnology and Neuromodulation in Trauma and Disease, which involves the development and testing of new technologies focusing on brain and spinal cord injury. We have exciting keynote speakers, featured talks, and presentations by our trainees who are addressing emerging themes in neural engineering. I hope you will enjoy this informative conference that continues to lead to new collaborations and ideas to move this exciting field forward. Once again, I welcome you to the Miller School of Medicine. Enjoy the conference. Back to you, Dalton.
Thank you, Dean Ford. That was fantastic. And Dr. Sarud Rajguru will be introducing Dean Biswas tomorrow morning. Again, a very important component of our vision for this particular institute. So now I'd like to introduce Dr. Patrick Ganser. Patrick is a new recruit to the University of Miami. And actually, it's the first dual recruit between the medical school in terms of the Miami Project and biomedical engineering. So really excited about doing this more in the future, bringing these scientists to be engaged in our neuroscience community as well as engineering community. Now, Patrick introduced me to a new concept, bioelectronic medicine, where he visions replacement of pills, for example, with electricity. So I think it's a really exciting time in this area. And now Patrick is going to introduce our keynote speakers for this morning. Patrick.
All right. Thank you, Dalton. And thank you to the Dean for the introduction. Thank you all for joining us today for the first day of the 5th Annual Symposium. I'll be introducing Dr. Michael Kilgard today, who is our keynote speaker. And if everyone can mute, I'm getting a little bit of echo. Dr. Kilgard is one of the world's leading scientists in the study of neuroplasticity. He received his bachelor's in molecular and cell biology from UC Berkeley and went on to do his PhD at UCSF. He is now the Margaret Fawn Johnson Professor at UT Dallas, also the Texas Instruments Distinguished Chair there, and also serves as the interim director of the Texas Biomedical Device Center. You'll be hearing about a bioelectronic medicine that Dalton mentioned, and really what he calls targeted plasticity therapy, or TPT, today during his talk. He's authored over 100 peer-reviewed publications in journals such as Science, Nature Neuroscience, and Neuron, receiving over $30 million for his research from organizations such as DARPA, NIH, Wings for Life, and CDMRP. You'll be hearing about some exciting clinical trial data as well to help patients with spinal cord injury during his talk titled Enhancing Recovery in Spinal Cord Injury Patients with Vagus Nerve Stimulation. Please give a warm welcome to Dr. Michael Kilgard.
There's something wrong with your microphone, doctor.
Yeah, Mike, you're quite scrambled during if you're talking now.
Yeah, I think he's talking. I think something's wrong with his microphone.
Yeah. Can anyone hear him? I cannot.
No, do you have anything to connect it to the audio that you're playing straight from the computer?
It might be a bandwidth issue. Perhaps if you stop the video and just do the audio, it might.
No, it was from before. It was from before. Even when the video was stopped, when he was giving the introduction, that's what came out before as well. And it came out from Dr. Kilgard.
I can log out and log back in.
Oh, now we could hear you. Yeah.
Okay, we can. All this is going to be edited, so just start like nothing happened. Introduce yourself.
Sorry if you said something prior. You're highly scrambled. Now it sounds great. Thanks.
Great. Excellent. Well, I'm really glad to be here. Thank you so much, Pat, for inviting me and the rest of the organizers of this meeting. It's a great day for research as more and more neuroscience and engineering collaborations are beginning all over the country. What I want to talk to you about today is what Dr. Ganser mentioned, which is a little bit about this idea of using neuroplasticity to treat patients with spinal cord injury. And I want to just step back and put the whole thing into context. I think all of us are aware from our education days that you can get better at just about anything. If you have sufficient practice and motivation, you're going to get improvement. And neuroscience has really helped clarify over the last 50 years that the improvements we make, whether that's on the playing field or the concert hall or in the classroom, is really all about synaptic plasticity. What we've learned is that practice over and over, going through drills or whatever it might be, is really engaging neural activity across millions of neurons. And these changes don't really occur unless you have sufficient motivation. And neuroscience has made it clear that that really depends upon the release of neuromodulators like acetylcholine and norepinephrine. So the idea has been discussed for many, many years that we might be able to treat neurologic and maybe even psychiatric diseases if we could take individual synapses and strengthen the right ones and weaken the right ones to improve symptoms for our patients. The real challenge is directing these new connections appropriately, trying to figure out how to rewire a human brain. It is no small challenge. Similarly, rewiring past damage in the spinal cord is really, really hard. So we've got a lot of techniques at our disposal, and over the last 10 or 20 years, there have been all kinds of additional technologies added to the traditional disciplines of occupational therapy and physical therapy. And we know how these tools work. They really are engaging the connection and formation of new synapses. And those synapses support recovery for our patients after injury. So the idea then is that rehabilitation is a form of practice. With sufficient motivation, our patients can make significant, meaningful recovery. Many people have studied this field and tried to understand how best to deliver rehabilitative training to improve recovery. And we know it needs high repetition, it needs to be progressive, starting doable and pushing harder and harder each day, individually tailored, task-specific, and requires a multidisciplinary team. The challenge, however, is that across a wide range of disciplines, we see that many deficits persist even after high-intensity, high-quality rehabilitation. So here's just one example of that. This is a recent paper from Catherine Lang looking at rehabilitation over 10 weeks in chronic stroke patients. And what you see is an upward trend. Here she was trying to investigate the role of varying the dose, how much intense therapy individual patients got. When you look at the raw data, you can see some patients did 3,200 trials, some did 10,000 trials. But what you see in each case is those people who start off bad end up bad, those who start off better end up better, and that the trends for the improvement are much smaller than the variability across patients. So people are not getting completely better for sure. And most surprising from this study is that there was no benefit whatsoever of tripling the dose of this therapy. So rehab is helpful, but more rehab is not more helpful, which is really quite surprising. So the key idea for this talk is that there's a simple neuroscience explanation for this observation, often called plateau. And that is that traditional rehabilitation strategies do not provide enough temporally precise neuromodulator release for optimal recovery. So let me unpack that a little bit. Some of these ideas may be new. There's a learning event. I gotta reach out and grab a ball or try to move my finger for the first time after an injury. And what we'd like to have happen is a burst of neuromodulators. We know from the work of Alfredo Kirkwood and others that when you get presynaptic activity, postsynaptic activity, and neuromodulators all co-occurring in time and space, you can form new synaptic connections. The challenge is really a psychological problem, which is that the brain doesn't want to change that much. And that when you go through hundreds and hundreds and hundreds of repetitions, what we see when we record in humans and in animals is that you really only get neuromodulator release during the first few trials. And after that, you rapidly adapt the amount of arousal, attention, and therefore neuromodulator release. So what we're really interested in is how can we find new tools based on our new understanding of neuroscience and our developing set of biomedical engineering tools to trigger release of acetylcholine and norepinephrine from the locus coeruleus and nucleus passalis. We need a safe way to do that. Ideally, we need to be able to selectively trigger it during moments of learning. So here's the idea, you're trying to form a new connection. Look at this great neuron. Wait a minute, I missed the point. I wanted to make that connection and it wasn't able to do so. If we had a label where we could say right here, go ahead and stop, make a new connection there, maybe we could have patients recover new function and improve their quality of life. So for my graduate work, I actually began using deep brain stimulation of nucleus passalis to release acetylcholine. And it's a really powerful, interesting tool to selectively engage release of acetylcholine and phasically pair that with specific events. You can get large-scale reorganization of the brain. The problem is the safety profile is not excellent. Many problems with implanting large devices into the brain, not least the cost, but also some significant risk. Many of you have heard of transcranial magnetic stimulation. Here's a tool that's known to be safe and can phasically, transiently release neurochemicals at an individual moment during a learning event. The problem is the selectivity is pretty poor. You're engaging simultaneously populations of millions of neurons at least, and that's not really the way you want to do it if you want to strengthen certain synapses and weaken others. You can get selective activation with stimulant drugs, nicotine, amphetamine. It's clearly safe, but it's not phasic. When you take a pill, it's going to be active for the next couple hours. So I want to tell you today about some new work we've made, beginning with a Nature paper in 2007, showing that vagus nerve stimulation is a surprising candidate to fill these three needs. It selectively engages the release of neuromodulators in a phasic, very transient burst activity, and it's known to be safe because 100,000 people have already received substantially more vagus nerve stimulation for the treatment of epilepsy. So here's the basic idea then. I'm going to do hundreds of rehab events, trying to move my finger, open and close my hand. Normally, I would get some release of neuromodulators and some neuroplasticity at the beginning. What we know from our animal experiments is that when we add neuromodulators, triggered by the half-second brief bursts of vagus nerve stimulation, patients aren't hurt, they aren't bothered, typically they don't even notice it, and now we can release neuromodulators as often as the person does these individual repetitions of their rehabilitative training. So the idea then is neural activity plus neuromodulator release leads to synaptic plasticity. We want the activity to be a form of rehabilitation. We want to increase neuromodulator release using vagus nerve stimulation, and we're hopeful that this additional synaptic plasticity will provide greater recovery than you could make with rehabilitation alone. So many of you have taken neuroanatomy classes and are aware of all these cranial nerves. Why would you stimulate this particular one, the 10th cranial nerve or the vagus nerve? I'll go briefly through some of the key advantages. First, there's just this excellent safety record. People are receiving vagus nerve stimulation now, 30-second trains every 5 minutes, 24 hours a day, 7 days a week for epilepsy. It's really surprising you can do that, not have horrible side effects, but that's the nature of the beast here. We're going to deliver about 100 to 500 times less stimulation than the FDA has already approved for epilepsy. 80% of the fibers are sensory. Most people think of the projections to the heart, for example, but really this is a sensory track coming up from the abdomen and the thoracic cavity, sending signals into the brain about what's going on in the body. And so when you activate these neurons, you can send a new novelty signal up into the brain. We're going to deliver very small amounts of current because we're only really interested in activating the large diameter stretch receptors, which are quite easy to activate. We've shown in animals, and a number of others have shown in humans, that when you do this, you trigger a very brief burst of acetylcholine and norepinephrine in the brain. It's about the duration of the electrical stimulation to the vagus nerve. In our case, it's going to be about half a second. And conveniently here, because the vagus nerve is coming up from the autonomic system, it's all subconscious. Most people do not notice it. If they do, what they're really noticing is a slight tickle caused by activation of the recurrent laryngeal nerve and a little bit of movement of vocal cords. So here's how we envision this tool to be used in the clinic. There's a therapist watching a patient do some form of physical therapy. In this case, he's lifting up a glass and putting it on an elevated platform. This particular individual in this cartoon has an implanted medical device underneath the collarbone that responds to the wireless signal that's sent from the laptop and activates this brief burst of vagus nerve stimulation, which is delivered in this case via a long lead up to the left vagus nerve, activating release of neuromodulators on both sides of the brain. So I want to tell you briefly by way of introduction about 3 clinical trials that have recently completed for a total of 145 stroke patients at the chronic phase, at least a year after their injury, and the benefits that they received by adding vagus nerve stimulation to their physical therapy regimen. So first, just a little bit about who we're talking about. These are all people who've had a ischemic stroke. They're in the normal age range, 22 to 80 years old. They all have hemiparesis, Fugl-Meyer scores from 20 to 50, but they do have to have some active movement of the wrist and a few of the fingers. Fugl-Meyer upper extremity subscore was the primary measure, but a number of other measures were included in these studies. So here's how the therapy is done. Subjects come in for 2-hour sessions 3 times a week, so 18 sessions over a 6-week period. We're trying to produce hundreds of movements per day, and the therapist might be requesting someone to reach and grab an object, but there's a whole bunch of things we do in a sort of standardized schedule, like flipping cards or doing simulated eating, putting objects into wells, or opening and closing bottles. Every individual is allowed to pick 2 additional tasks that they want to use to increase their motivation. One person wanted to do stapling and fastening and unfastening his belt. Whatever the task is, each time they perform the task, the therapist pushes a button and activates a brief burst of vagus nerve stimulation paired with that particular movement. In this first study I'm going to tell you about from 2016, published in Stroke, there were 2 groups. We randomized subjects to either do rehabilitation with vagus nerve stimulation or rehabilitation alone. Again, it's just a half-second burst. And in this case, we randomized who was to receive the implant. So here you see the improvements over these 6 weeks of rehabilitative training. And what you see is a significant improvement in those who had vagus nerve stimulation. Now, initially, we were excited about these results because they mirrored our earlier animal studies. But obviously, in an open label study like this, it's easy to tell who's got the vagus nerve stimulator. And so you worry that these improved recovery is really just due to increased motivation for the people who can feel that they've got a device in their chest. So we repeated the same study, except this time everybody got the implant, but we only turned it on in half the individuals. We were very pleased to see the same result, which is a statistically significant improvement in recovery for individuals who had vagus nerve stimulation compared to those who got a sham amount of stimulation, sufficient to convince them that they were receiving active stimulation. In this case, these people have got an implanted medical device that's never really been used. That seems like an unethical study, except that, of course, you can cross this group over, have them repeat the same 6 weeks of physical therapy, this time with the vagus nerve stimulation active during each and every one of their movements. And now you see that the same subjects make gains that they did not make during the same therapy delivered with the same therapist just 6 weeks earlier. So this is consistent with this idea that vagus nerve stimulation increases neuromodulators and helps people make gains they couldn't otherwise make. It was sufficient for the Food and Drug Administration to approve a pivotal trial that recently completed. They tested 108 patients across 15 sites, and the basic experimental design is shown here. So we consent our subjects, we do baselines before and after implantation. They're randomized, everyone's implanted, but they're randomized. The sham group gets just 5 bursts at the very beginning, a little bit of a tickle to think something's happening. And the others are getting vagus nerve stimulation during each trial. Then there's an assessment, and then there's this delayed start group where the group that got rehabilitation only at first crosses over to receive active vagus nerve stimulation. So here's the results of this study published in Lancet a few months ago. Again, a highly significant improvement in the upper extremity subscore for the Fugl-Meyer stroke assessment. There were improvements that were highly significant on many other measures, including the Wolf Motor Function task. Here we had 57% responders with a clinically meaningful improvement compared to only 22% in the control group. So this means the number needed to treat is less than 3, which is quite good for a medical intervention. The only serious adverse event out of 108 people was one case of vocal cord paresis, presumably due to damage to one of the nerves during the implant, but it resolved fully within 5 weeks. So as a result of this excellent safety and efficacy data, the Food and Drug Administration approved this new therapy for chronic stroke patients using the spin-off company from UT Dallas called Microtransponder. The system is called the VivaStim Paired Vagus Nerve Stimulation System in August, so just a few months ago. And it's approved to treat moderate and severe upper extremity motor deficits associated with chronic ischemic stroke. So what I want to go over today is kind of how we're transitioning this technology from initial studies in animals all the way up to our ongoing trials in individuals with spinal cord injury. So the first 4 questions we're going to address before we get to the clinical data is really who might benefit from this? Why would you think this could work? Stroke and spinal cord injury are really quite different. What rehabilitation should be delivered? When should you deliver vagus nerve stimulation? How much? So these studies began quite some time ago, almost a decade ago now, in animal models where we can produce really precise lesions. We began with endothelial lesions of the cortex and later of the subcortical structures as well to produce an animal model of stroke. With our engineering colleagues, most notably Rob Reneker, we were able to develop booths where the animals can participate in fully automated motor assessments. So here you see just one of our racks. It's a dozen animals simultaneously reaching out, grabbing a handle and pulling it. If they pull above 120 grams, a third of their body weight, we would deliver a pellet from that rack of pellet dispensers across the top. On the left, in the upper right screen, you see the vagus nerve stimulators. We can have a wire that goes to the top of the rat, connected to an electrode on the left vagus nerve, deliver a half-second burst with each successful trial. So here's the basic experimental design. We shape the animals in this task, make sure they're really good at performing it, then give them a stroke, let them recover for a while. We assess them to make sure that they are still able to do the task, though they are clearly impaired. We randomize them to receive intensive rehabilitation or intensive rehabilitation plus vagus nerve stimulation. Continuously throughout the trials, we're measuring the forces that they're producing and delivering vagus nerve stimulation in a closed loop manner. So only when the animals are pulling and in this case, only when they're pulling at above average levels is a brief burst of vagus nerve stimulation delivered. So here you see the animal's performance on this task. Almost 90% of the time, these animals can reach out, grab that handle, and pull sufficiently hard to cross our threshold. After this lesion to motor cortex, you'll see the animals have a significant impairment. When they do physical therapy, it's no surprise, they get improvements, statistically significant improvements, but they have remaining deficits even after many, many weeks of physical therapy, consistent with what we see in our patient populations. When we add vagus nerve stimulation to randomly assign half the animals, in this particular animal model, they made a complete recovery and were returned to pre-lesion levels. Vagus nerve stimulation was only delivered during therapy and only for the first 5 weeks. During the 6th week of therapy, there's no vagus nerve stimulation at all, demonstrating that these effects are very long lasting, consistent with the hypothesis that they're caused by new synaptic connections that support this motor recovery. Now, those animals were young, and it was relatively short period of time between the lesion and the implantation. So here on the left is a study looking at animals who are aged. On the right is a bunch of animals who had waited more than a month after the lesion before delivering any vagus nerve stimulation. And in both cases, you'll see the same thing. Rehabilitation is helpful, but rehabilitation with vagus nerve stimulation provides additional gains that cannot be produced with rehab alone. We've tested other conditions, including on the left, hemorrhagic stroke, and on the right, traumatic brain injury models in animals. And again, it doesn't seem to matter the specific mechanics of how you damage the motor system, but that there be some impairment and that rehabilitation be effective but incomplete, then vagus nerve stimulation appears to be therapeutic. With support from Wings for Life in Salzburg, we were able to secure a grant to test the hypothesis that vagus nerve stimulation might also help in animal models of spinal cord injury. Pat was willing to come over and join this team to try this really audacious project. We were really quite open to the idea that it might not work for spinal cord injury, that it might be specific to brain plasticity, and it would be too difficult to make changes down in the spinal cord. But as you see here, what Pat was able to demonstrate was, again, intense impairment caused by spinal cord injury, whether it was a lateral contusion model or a midline contusion of the C5. Intense physical therapy is good for you. Animals are highly significantly improved, but they remain substantially impaired. When we added vagus nerve stimulation, you see that were statistically significant improvements all across the board that persisted long after vagus nerve stimulation ended. Moving on, wanted to test whether or not, before we went into patients, maybe this was specific to C5. What if it went a little higher, a little lower? Oftentimes we go to the clinic too quickly. We don't really understand which patient populations to select. So we were worried, and in fact we predicted that if the primary motor neurons themselves were damaged, in this case the C7 area has a lot of the motor neurons responsible for forelimb grasping, that there'd be no hope of recovery. What we saw instead was a really surprising result, that in fact vagus nerve stimulation continued to benefit these animals even when the lesion was done at the level of C7, right in the middle of their motor neuron pools. And our hypothesis is that although we're hit in the middle of the motor neuron pools, some areas above are spared. Some just below are, of course, obliterated. The animal model here is a incomplete injury model for sure. And so the idea here is it doesn't really matter where in the anterior posterior extent of the cord the damage is done. What's most important is that there must be some spared pathways. That was the hypothesis to explain this surprising result. We have gone ahead and tested this now in 9 different animal models, including models in peripheral nerve injury. In each case, what we see is so long as there's a pathway, the brain can find a way, but the brain needs our additional help. Evolution did not prepare us for recovery from severe events like stroke and spinal cord injury. And so we don't really have the best tools we possibly could, but by adding this vagus nerve stimulation, closing the loop, we hypothesized that we could provide this additional signal that the brain appears to be missing. So how can we improve performance? Is it just the task that you perform, or can you do it and generalize to other tasks? So here's an animal model where the animals are learning now, instead of to pull that handle, they're turning a knob. And so they got to turn 75 degrees to get their pellet. They're able to do it after a stroke. It's really hard to do pronation supination, as it is in many of our patients. And what you see here is, again, rehabilitation is good for you, but rehabilitation with vagus nerve stimulation is better. These animals have never done the pull task. And one day we added the pull task without any vagus nerve stimulation to look at generalization. We want our patients to be able to perform any range of volitional goals with their hands after therapy. And so here the therapy has ended on week 5, and you're still seeing significant improvements on a brand new task on week 7, 8, 9, and 10. And even when you wait until week 11 and cross them back to the supination task that they've not done for more than a month, you see that these gains are still maintained. So how do we figure out what vagus nerve stimulation to do? This is really a challenge. It's very important to set the parameters just right. There's an inverted U. All things in moderation is what Ben Franklin said, and we see that here as well. If you stimulate too much, there's no effective improvement in motor cortex plasticity, which is what this study was done in healthy animals. The same thing is true of frequency. If you do frequencies are too high or too low, there's no neuroplasticity produced. If the duration is too long or too short, if the interval is too fast, we also see that the benefit begins to be reduced. All of these are consistent with this idea that we're trying to engage a neuromodulatory system that is trying to determine when to engage changes and is very selective, very picky about when it wants to make these new changes. So how are these changes occurring? I've claimed that these are due to synaptic plasticity, but I've not yet shown you evidence. We don't have evidence yet in our human stroke patients or spinal cord patients, but we do from our animal studies. So here's a study where the animals again receive a lesion, again they're impaired, again rehab helps them, and again physical therapy with rehab is better than physical therapy alone. But in this case, we've lesioned a handful of neurons, in fact, just a few hundred neurons that are projecting from nucleus basalis to the motor cortex and releasing acetylcholine. Without that handful of neurons that are releasing acetylcholine, there's no benefit of vagus nerve stimulation whatsoever. We know that the timing is critical because we have delayed the delivery of vagus nerve stimulation, so the animals in black have an intact nucleus basalis projection, and they also get vagus nerve stimulation. They also get physical therapy, but the two are offset by about 2 hours in this particular case. In Pat's study, they could be delayed by as little as 20 or 30 seconds, and there's no benefit whatsoever. So even when you're interleaving vagus nerve stimulation with active rehabilitation, no benefit unless it's delivered specifically during the most effective movements. So what are the mechanisms that support this recovery? Again, animal models allow us unprecedented precision to be able to explore down to the millimeter, in fact, the micron level, what's going on in the brain. So at the millimeter level, we can map the motor cortex. You insert microelectrodes into motor cortex, and if it's left motor cortex, when we stimulate the right spot, you'll see the paw close or open, or the hind limb move, or the jaw move. We can make an entire map of the motor cortex representations. Using a tool that Pat introduced to our laboratory, pseudorabies virus 152, you can inject transsynaptic labeled viruses into the grasping muscles and then literally count the number of neurons in the brain that are connected to that particular pathway and look in a totally hypothesis neutral fashion at both sides of the brain and normalize all the data based on the amount of virus that's been injected. So first, the motor cortex plasticity. We hypothesized we were producing neural plasticity. We had shown it in healthy animals. And here you can see in our stroke model, our nerve transection repair model, and our spinal cord injury model. In all three cases, we see a reduction in the number of motor cortex neurons that produce active movement of the forepaw and an increase with vagus nerve stimulation paired with physical therapy. So this suggested that there was some new pathway even in these anesthetized animals, but where exactly are these new connections being formed? With the pseudorabies virus technique, you can actually see where those synapses are. They must be formed in the connection from the motor cortex to the spinal cord. So these corticospinal synapses is what we can count basically by looking for labeling of the pseudorabies virus marker, green fluorescent protein in the brain. So here again, we've doubled or tripled the number of neurons in motor cortex that produce these movements. We also doubled or tripled the number of connections between motor cortex and the relevant muscles here. One of the most beautiful things that Pat was able to accomplish is really to help us understand in our different animal models, how can it be that you damage different pathways and the animals still recover? So here what we see in the lateral contusion, the sort of Brown-Séquard kind of model, we spared the corticospinal tract, which runs down the midline in the rat cord, And you see, again, improvement in motor cortex connections without any improvement in the red nucleus or propriospinal pathways. Counting these cells is like Christmas trees. You can't miss them. They're a really obvious label. There's nothing really subtle here. When we switch to the central cord syndrome model, now the corticospinal tract is basically obliterated, but because we hit the middle, we've left the lateral pathways of the propriospinal tract and the rubrospinal tract. And here again, we see doubling of the number of connections there, presumably supporting these connections. And again, because we can count these individual cells, we know exactly the relative proportions of connections from the primary motor neurons back up to the motor cortex. So that's a lot of progress with motor pathways, but that's not the end of the story for spinal cord injury or for stroke. There's a lot of sensory loss that occurs with these conditions. So we were interested in whether or not vagus nerve stimulation during tactile therapy could improve sensory processing. Before we ever had the chance to do this in animals, we had a subject who snuck through our exclusion criteria. We intended to exclude people with profound sensory loss, but one participant had actually pretty good motor function, was already implanted, received some benefit from vagus nerve stimulation during therapy, but it was clear that his real deficit was really sensory in nature. So blindfold the participant, do passive stimulation, in this case with a paintbrush, pair it with vagus nerve stimulation, or active haptic exploration. Here he's trying to feel a coffee cup, and see if by pairing vagus nerve stimulation with sensory responses, this person could get better. We suspected that might be possible because we knew in the auditory system from our earlier Nature paper in 2007 that pairing tones could rewire the auditory pathway. We hadn't done anything with somatosensory pairing yet. What we see here for this one patient was a significant improvement in tactile thresholds within just 10 days of therapy that were persistent long after therapy. His diagnosis went from chance performance 10% for 10 objects and gradually improved throughout his 4 weeks of therapy, reaching a final result of 70% correct, identifying everyday objects. Percent correct for joint position, so proprioception improved significantly, though was not returned to normal, and even responses to deep and light touches improved. So here's an N of 1 person. People have seen lots of interesting case studies in both stroke and spinal cord injury. So we decided to go back to the animal model and see if we could recapitulate in an animal model of sensory loss whether or not vagus nerve stimulation could indeed improve sensory function when paired with tactile stimulation. So here's a bunch of animals who are withdrawing from a poke to their paw at 8 grams after a nerve transection, a median and ulnar nerve with repair. They recover their ability to feel, but they have poor function. So it requires about 4 times more tactile stimulation for them to withdraw. And these deficits are very stable across the rest of the animal's life. When we added vagus nerve stimulation plus touch to the paw, we see the animals rapidly over a number of weeks improve their tactile thresholds in a way that tactile stimulation alone does not produce. And then after vagus nerve stimulation ends, we follow these animals for a couple of months, and we saw that these improvements seem to be lifelong. So how are we going to deliver tactile therapy in the clinic, let alone at home? We think we're going to need thousands of repetitions. We developed a new box that we're in the process of using with our spinal cord patients. Here's a healthy subject, actually the inventor of this technology, producing passive haptic exploration and stereognosis training. So there's a curtain so the person can't see it. The camera is looking down on the objects and picking out what color you should be finding and adding back in. So you're supposed to be adding a yellow object. You did add a yellow object. That's how it knows you got it right. In this case, she's telling objects that are different sizes. She's presumably using their length, but she could be using their width. But you could make these anywhere from really easy for individuals with severe impairment all the way down to really hard, near 10% difficulty levels. Are easy for us to print with a 3D printer, but very difficult to discriminate actively. And we're not limited to size or shape. We can do different shapes, different textures, even different weights where the objects are exactly the same size, but they've got differing amounts of hollowness. So how many repetitions are we going to need to pair vagus nerve stimulation with therapy? The rats, at least for motor, are getting hundreds of repetitions. And in our first stroke patients who did 6 weeks of therapy, they were also doing hundreds of repetitions per day. We had reasons to believe that we would probably not be getting the maximum gains in only 6 weeks for a human. And so we wanted to be sure we could deliver therapy to people even with very severe impairments, and then eventually be able to send these therapy mechanisms home. So we developed a suite of tools called Recheck, where we can measure the pinch force, the wrist force, but also range of motion for pronation, supination tasks, and the like. These are very sensitive measures with modern strain gauges. You can see, of course, that individuals with spinal cord injury, shown in red on the bottom, are about 10 times weaker in terms of their pinch force compared to healthy individuals. But even that 10 times weaker is easy to detect with a strain gauge. And so if you put someone's fingers in, you can have them engage with this task and do many, many repetitions. We're measuring in real time what that signal looks like, whether it's with the range of motion task here or with the isometric tasks. We've shown that for GRASP, the best developed upper extremity assessment for spinal cord injury, there's a highly significant correlation between how well you do on this battery of fully automated assessments compared to how you do with the clinician-administered GRASP assessment. Now the challenge is sitting and repeating opening and closing your fingers is again pretty boring. How are we going to increase motivation? So here you see our first subject actually in this trial doing a simple breakout like Pong game, and she's pinching and opening her fingers to control the left and right movement of that little paddle at the bottom. And this means that it's a little bit less boring, a little bit more gamified, and we'd like to make it where patients can go home with this therapy. So she actually took this set of tools home last week. For each subject, we write an individualized prescription for that particular person based on the deficits we document in their clinical assessments. And so we can tell them how many movements, how many minutes, and we can adjust the gain so that they are challenged in terms of how hard they have to squeeze the puck or how fast they've got to move between the objects or how many keys they've got to press per second on a typing exercise. Again, to adjust it individually to skills that they need to improve and muscles they need to strengthen. What's nice about each of these tasks is all of them are streaming the data to disk. So if you're taking an object like this accelerometer, gyrometer puck with a strain gauge made by Flint Rehabilitation, as you turn it, the gyrometer and accelerometer know that that's being done, and so we can look at the angle of the puck over, in this case, 60 seconds of data collection. But you could be rolling the puck, or you could be typing keys. In each case, this is just, again, a very brief 60 seconds of data, but we can do an hour and a half of data for each patient and receive basically continuous assessments of what their performance looks like. We know that subjects, at least with spinal cord injury, stroke and brain injury, are able to do these things at home. And so here they are first in our control conditions in the clinic doing several hundred, and then we send them home and they're able to do even more than 1,000 trials at home. As expected, these people are find these games to be more challenging, set to the same levels as an individual without impairment, but they are able to do them because we've adjusted the gains to be appropriate for individuals with stroke. So when they're doing fruit archery, they hit fewer pieces of fruit per minute, or if they're swiping their finger across a touch screen to do Fruit Ninja, they're not as accurate, but they can engage with these tasks and they find it to be satisfying and rewarding for them. So here you see just one of the tasks, again, where the subject is, we're measuring both the strain gauge as he pushes with his thumb, as well as the pronation supination angle as he turns the puck to aim at each of these pieces of fruit. So now the next question is, how are we going to deliver this to the vagus nerve? We're really excited about the possibility of using non-invasive techniques. Many commercial manufacturers have come out with tools that they purport to be able to activate the vagus nerve through the skin. Unfortunately, we did a bunch of animal studies, and it's pretty clear that you can't remotely get to the vagus nerve by delivering current through the skin, mostly because it'll be painful well before you activate the vagus nerve. So we showed in animal models, of course you can, if you deliver enough current, you'll eventually activate the vagus nerve, but the nerve threshold is about 10 times higher when you're activating it through the skin than it is before you start to reach the pain thresholds when the muscles get activated. So we're pretty confident that normal methods of transcutaneous activation are not actually activating the cervical branch of the vagus nerve. So we've been focused on developing a minimally invasive technique. If you can't do it non-invasively, you may as well do minimally invasive. The trial I mentioned before, the microtransponder Vivistem device, used a large implanted medical device under the collarbone with a lithium-ion battery and a long lead up to the nerve. Rob Reneker and the engineering team here at the Texas Biomedical Device Center has developed a next generation technology. It's about 50 times smaller and sits directly on the nerve itself. So it's cheaper for us to make. It's completely wireless because the leads tend to break on devices that move back and forth. It's less invasive, MRI compatible, and fully programmable. A few months ago, the Food and Drug Administration announced that this Restore device, the one on the left, was awarded breakthrough device designation. So it is not yet approved. The one on the right is, but we're on an expedited regulatory pathway to get it toward approval. We can make lots and lots of these devices because we use modern manufacturing wafer technologies, not hand production and soldering, as is used in most medical devices these days. So here's what the tools looks like. The device is sitting there on a small coin. It's held in place with a little silicone cuff with a little zip tie sort of technique. It's powered. The reason it's so small is there's no battery inside, so it's powered by the coil you see on the right, which is held in a high-impact plastic housing and then positioned over the device with a soft neck band. So how are we going to integrate these whole things? Well, originally we did it with a physical therapist, engaging hands-on with the patient, in this case spinal cord injury, for, again, 6 weeks. And the therapist has got a cell phone. That cell phone has a button on it. It sends a Bluetooth signal to the power and control module, which activates the device implanted in the skin, under the skin, and engages neurons, in this case in the motor pathway, because they're doing a motor task at that time. We can also do this in a fully automated way. We've just received FDA approval to do this at home in individuals where all the stimulation is not delivered by the therapist, but by a simple algorithm that picks the peaks and delivers VNS during peak performance. So you see here just an example therapy session for one participant, and we can go through picking any individual section and select out when are the above average trials for this particular person and how can we deliver vagus nerve stimulation time with them. That's pretty easy for a computer algorithm to do. So sort of wrapping up, we've added to this idea that we want high repetitions, progressive, individually tailored, task-specific therapy done by a multidisciplinary team. We think we have evidence in both humans and animals that adding vagus nerve stimulation helps, that we can deliver vagus nerve stimulation in the clinic and at home, and that using this fully automated therapy will allow us to better document what exactly it was that were part of the active ingredients for improving recovery in these subjects. So the trial that's ongoing now is an early feasibility study, but we decided to go ahead and do it double-blinded, placebo-controlled. So we recruited a bunch of people to ask these questions. Is the device safe? Does VNS improve recovery? And does more VNS improve more recovery? So provide more recovery. So here, during the double-blinded first 6 weeks, half the people are getting active VNS and half are not. But everyone goes and does another 6 weeks. So not just the group who got sham therapy. So the group on the left is going to get 12 weeks of VNS. We wanted to ask the question, is the second 6 weeks adding additional value on top of the first 6 weeks? And the answer, I think, is going to be yes. And the issue on the right is helping us determine, is this really due to VNS or is this some placebo effect? We've just submitted to the FDA to go ahead and go beyond this because our initial results look like 12 weeks is better than 6 weeks. Why not 50 weeks? When does this plateau? So we're asking for permission to do more than 100 sessions to see if we can just continue to make gains over a long period of time. So the trial we're running now, we're selecting for people with cervical chronic spinal cord injury caused by trauma more than a year ago. They need to have some residual hand and limb function, 18 to 64 years old. Our primary outcome measure, of course, is safety, but we're using the GRASP assessment as well as a whole battery of other assessment tools to look for improvement in these patients. Here's our progress to date. We're approved to do 20 patients. One, patient 5, was too impaired after assessment. But you'll see we've completed the blinded phase for 4 of the participants. 2 others are in the middle. 2 have started a few days ago, and 1 more starts actually in about 2 hours. So here's the data from the first 2 participants to finish the double-blinded component. You'll see on the left, those that were randomized to sham had no improvement in their GRASP assessment of upper forelimb function. Those on the right both received clinically meaningful improvements. And most notably, they're receiving just as much gains in the second 6 weeks as during the first 6 weeks. Because we're assessing every movement, every minute of every day during therapy, we can actually track these individuals' performance, in this case on their pinch force as they're playing video games, whether they're driving a car or breaking those little blocks. We can see exactly how well they're doing. Of course, it's noisy. Some days are more tone than others. The second subject is the red one, actually improved by more than a factor of 10. Our first subject is that one in blue, where you're talking about a little over a doubling of her pinch force. So we're waiting for much more data, obviously still to be determined what the result of this trial is, and a lot of work to eventually expand this to multi-site clinical trials and test this in a sufficient number of people to actually determine whether or not vagus nerve stimulation could be effective for the treatment of chronic spinal cord injury. But I think the compelling set of data from animal models and now from humans does confirm this basic idea that traditional rehabilitation alone does not provide sufficient temporally precise neuromodulators for optimal recovery. We can do better. Vagus nerve stimulation is a safe and powerful technique to deliver temporally precise neuromodulator release and enhance rehabilitation of motor and sensory impairments caused by not just spinal cord injury, but other impairments to the central and peripheral nervous system. Automated rehabilitation techniques has the potential to reduce the cost and improve the quality and amount of therapy and allow it to be delivered at home. I want to thank everyone at the Texas Biomedical Device Center, most notably Seth Hayes and Rob Renneker, who really helped me lead the center, especially as we're trying to do so many things with a small number of people, iterating a cycle between science leading to engineering development, going into the clinic and testing them, and iterating back through the animal models to develop the next generation of engineering tools. I want to acknowledge the funding sources, most notably in this case, the Defense Advanced Research Projects Agency and Wings for Life that are paying for the spinal cord trial. Also to acknowledge my potential conflict of interest. I'm a consultant for and shareholder in this little spin-off company that came out of UTD called Microtransponder. I want to remind everyone that the therapies that are described in this talk are not FDA, with the exception of the ViviStem device that just received FDA approval a few weeks ago. And then most notably, I want to acknowledge Pat's incredible effort to this. We just absolutely would not be here without his willingness to try this really high-risk project. And then our clinical collaborators, Jane Wigginton, our Chief Medical Officer, and our two close collaborators at Baylor University Medical Center, Rita Hamilton and Chad Swank. With that, I'd be happy to take any questions.
Do we have any questions? Any questions?
Sure, I'll ask one. Thanks, Mike. That was a really great talk. Maybe these are kind of basic questions, but I was wondering, how do you actually measure neurotransmitter release in people? Are you using something like pupil dilation for that, given that's given the LC projections? And then a related question, given the relative non-specificity of LC projections, presumably vagus nerve stimulation could be a means for therapy for a whole host of different conditions across the sensory modalities.
Yeah, no, that's a great question. So in the animals, of course, it's easy. You just take an electrode and stick it in locus coeruleus. It looks like someone just flipped optogenetics on it, and the neurons just fire like crazy, and then they stop as soon as it ends. So you can't miss it in animals. In humans, of course, it's much more challenging. A number of studies have been done using fMRI and all the rest, but it's a pretty tiny target. So I would say consistent with, but not definitive evidence. In humans, you certainly can get pupil changes. They're really pretty subtle. And in fact, we have trouble looking for a good biomarker because mostly people don't notice anything. You don't get heart rate changes. You don't get reliable signals. A new study came out this week, actually, looking at when people are sleeping and getting VNS, and you get changes in EEG desynchronization caused by these. That's probably the best way to do it. But it's a pretty subtle effect. And that's why you need to do it lots and lots of times. You don't do this 5 times a day. You do this hundreds of times. So you're really sort of biasing the system, helping that gradient descent using tools like sort of helping someone solve the credit assignment problem. Which synapses are the ones that are making this happen? Well, you're going to need a lot of statistical evidence. And so, yeah, you're bathing the brain in acetylcholine and norepinephrine, but what is the area that's most consistently happening with it? A handful of neurons in motor cortex, what's really driving this activity. And the brain appears to be exquisitely sensitive to making those statistical analyses. So that's how we think it's engaging. That's where we think the specificity comes from. But you're right, we started in the auditory domain, we paired tones, and we got massive changes in the auditory system. So we're always worried, like, well, we wouldn't want to change auditory systems, people got spinal cord problems. So we try to make sure that nothing else is correlated except the thing that we want to be. But you're right, we're looking for kind of how far could you go. Just to update you on some other ongoing trials, we've done a lot of work in animals on post-traumatic stress disorder, and we've had 2 patients now with very severe forms of PTSD who receive VNS paired with a different form of therapy. In this case, that therapy is prolonged exposure therapy, where they sit and imaginally rehearse their therapy, and then they record it and go home and get VNS at home. Neither of those first two patients have post-traumatic stress disorder anymore after more than 10 years of suffering. So again, that's an open label study, don't have any severe data, but yeah, sensory function, motor function, cognitive function, really synapses do everything in the brain. If it's behaviorally mediated, probably synapses are what's causing it. And we just need a new technique to get us down to that level, which is much lower than the brain area, much more specific than a cell type, really down to the individual synapse level.
Mike, that was an outstanding presentation. Thank you very, very much. So safety is always a concern. Have you seen any risk factors associated with vagal nerve stimulation? And when you really start targeting the sensory nervous system, some people, especially a lot of patients living with spinal cord injury, already have neuropathic pain. Have you seen any evidence that it alleviates pain or aggravates pain or anything like that? Thank you.
That's a great question. These are absolutely things we worry about the most. So let's talk about the comorbidities of people with spinal cord injury, things you might worry about. Obviously, FDA was greatly worried about things like us triggering autonomic dysreflexia. That would be really bad and potentially dangerous. So in collaboration with Andrei Krasikov, we were able to complete a series of experiments in animals where we did spinal cord injury and then intestinal distension to trigger autonomic dysreflexia, add vagus nerve stimulation specifically during an event. We thought it might help. The answer is it doesn't do anything. So it doesn't exacerbate it. It doesn't make it better. It just didn't do anything at all. So that got us over that worry. And then we were sort of trying to figure out, well, what else would we be able to address? We've done a number of experiments in chronic pain. Obviously, this is an enormous condition, one of the most common and debilitating symptoms of neurologic injury. It's interesting. People often ask, is this good for everything? And the short answer is, to date, no, it's not good for everything. It doesn't appear to be good for pain. We can't figure out why. So in principle, rewiring the brain ought to be able to alleviate pain. If you just did long-term depression on all those circuits that are hyperactive, you should have less pain. It's a simple hypothesis. You could use gate control to increase the responses to the suppressive responses or decrease the responses to the pain-producing responses or change the rhythmic circuits in the brain. Any of those are possibilities. We've tried it a lot of ways, and thus far, none of them worked. So we're now in the process of reporting. It doesn't always work. If you pair the wrong stimulation activity pattern with vagus nerve stimulation, it doesn't help. We're still looking. There may be some new way of doing it, some alternative way of engaging the system that might drive the right kind of plasticity. We haven't found it yet. We've also done experiments in hearing loss, looking at maybe we could strengthen the response to impaired hearing. And so far, we're about to publish again. Doesn't appear to really help. We could drive plasticity, but plasticity alone doesn't help. You've got hearing loss caused by hair cell damage. You're missing 90% of your hair cells. A little bit of plasticity is not going to solve it. So we don't want anyone to think this is a magic wand, you just get the neural plasticity and everything's better. This is a really hard problem. People are significantly improved. They still have lots of disability left. And so we've got our work cut out for us to try to figure out how to better deliver therapy and how to optimize the delivery of vagus nerve stimulation to drive the right kinds of plasticity. But we don't yet know how circuits lead to behavior. And so it's a little bit of a challenging problem. Now that we have this tool in people, we think it can really help us better understand what needs to happen and move forward in a sort of virtuous cycle for better therapeutics.
I had a question. That was a really great talk. I was wondering about the mechanism of this effect. Do you think that it is via the neuromodulatory changes in responsiveness to different types of transmitters like glutamate and so on and so forth? Or do you think the mechanism is related to facilitation of regeneration?
Yeah, it's a great question. Vagus nerve stimulation does so many things that it's sort of hard to parse out what exactly is the most important thing. Here's just a list of published things that it's known to do. I mean, it actually produces neurogenesis. It's sort of amazing. It certainly reduces inflammation, and there's any number of effects that have been well documented, from brain-derived neurotrophic factor to cadherins and voltage-dependent calcium channels. So in terms of what it engages, everything involved in synaptic function, learning, and plasticity are engaged and have been published to be engaged. What we know is that there's an inverted U, so activating more of these things doesn't help. So you kind of got to get the sweet spot, and we're still trying to understand better. Why is it that more is not better? You normally think more activation would be helpful. So I think the answer is certainly all of the known molecular biology that relates to synaptic plasticity. That's actually my background. All those things are actively and efficiently being engaged. The key really is the temporal specificities occurring because patterns of inputs, pre and post timing, the sort of Hebbian things, alone don't do anything. Hebbian learning doesn't do anything unless you've got neuromodulator. If you take a drug that blocks acetylcholine, you get no plasticity at all. The big surprise to us is how these things work in a cocktail. This is just mind-blowing to me. But if you lesion acetylcholine, no plasticity whatsoever. If you leave acetylcholine intact, but lesion locus coeruleus, no plasticity at all. So then we did this control experiment. Let's lesion the dorsal raphe. We also blocked it. So now you say, this is crazy that you need the combination of a soup of these neuromodulatory systems. But in fact, this is exactly what Alfredo Kirkwood at Johns Hopkins predicted, that really the spike timing dependent plasticity rule, this causality determining symmetry producing mechanism in the brain that helps synapses know which things to strengthen, which things to weaken. They don't even get created until you have this cocktail of neuromodulators. So the four humors idea that acetylcholine does this and norepinephrine does that, that's not right. They're somehow working in some kind of a soup. It's just incredibly difficult to work through.
Are the serotonergic systems engaged? Have people recorded from putative serotonergic neurons or measured release of serotonin in target areas with VVN?
They absolutely are.
Yes.
So Manta 2009 did a study looking at that activation and release of serotonin. We did studies showing that when you block those things, you don't get the plasticity effects. So serotonin is definitely involved. New study just came out again from UTD showed dopamine is actually not engaged, which is one of these rare things. Where's the dopamine? It's not rewarding. You don't get place conditioning in animals. So dopamine doesn't appear to be it. It's just more of this arousal, learn it kind of a signal than a motivation valence kind of a signal.
Thanks.
All right. That was a fantastic talk, Dr. Kilgard. It's exciting to hear all the preclinical and clinical progress. Hopefully you'll be able to join us for the panel discussion later at noon Eastern time, and we'll have lots of time for more of this kind of discussion and more questions as well. So to keep on track, we will now proceed to the student presentations, which are basically videos overviewing some of their interesting neural engineering research in a poster format. I will pass it. I don't know if Dr. Prasad, if you have any comments before we get into the next section here.
We just have a quick question in the chat. I don't know if we have a couple of minutes to address that. There is a question which says that, was there any VNS deficit in stroke patients? And we can move on to the student presentations right after that.
I'm sorry,
So
I didn't quite get the question.
The question is, was there any VNS deficit in stroke patients?
No, there was. I'm not sure what you mean by a vagus nerve stimulation deficit. So they were able to receive VNS the same way as every other patient. They received the same parameters. So there wasn't any reduction there. The side effects, there was no reduction in performance or anything like that. So I don't know of any deficit in vagus nerve stimulation in the stroke patients. They were improved in their recovery. Again, they're still disabled. It's not like this is magic by any means, but it didn't create any adverse event for them.
Okay, thank you. Yeah, we can definitely come back to this during the after we are done with the talks. So we can move with the student presentations. We have four videos after this. So waiting for Iggy.
Brain-computer interfaces have great potential in restoring independence to individuals living with paralysis. Surveys for individuals with quadriplegia have identified that arm and hand function hold the highest priority in comparison to other lost functions. In the laboratory, BCI research studies have shown some remarkable achievements, including communication restoration through speech synthesis and fast texting. Despite this research, these advancements have been limited to the laboratory environment. Invasive systems using microelectrode arrays often require recording rigs that are too large for portability. There have also been many attempts to place non-invasive systems into the home environment, but these systems have setup times that are too complex for practical use. These studies have provided valuable insight into potential BCI user preferences, which include system performance, functionality, and ease of configuration, without which BCI device adoption will ultimately fail. We recently deployed an invasive ECOG-based motor BCI system to the home of a patient with cervical spinal cord injury, and we are now analyzing these metrics of performance and ease of use after a year of home deployment. For this interface, we placed 4 pairs of electrodes over the subject's sensory-motor cortex, which provided us with 2 time series channels sampled at 200 Hz and 2 power channels sampled at 5 Hz. With a battery pack and a mini computer attached to the back of the patient's wheelchair, a mobile phone application for the user interface, and a motorized glove for hand grasp, we deployed the system home and used it for collecting data to train a decoder. The mobile phone application presented move or rest prompts to the patient. At the same time, we would record data from these channels so that the signals would be labeled with the motor imagery. Each data collection trial lasted for 5 minutes, and the prompts alternated back and forth at random length intervals between 6 and 10 seconds, and 33 of these trials were collected for decoder training. We then windowed this data at widths of 3.2 seconds and computed power spectral estimates using multi-tapering for the time series channels 1 and 2, and we used the Windows median power for channels 2 and 4. Averaging these spectral estimates revealed the differences between the 2 motor imagery states, particularly within the beta frequency band. And aggregating these metrics provided us with 644 features to use for decoding. The decoding architecture took these features and converted them into a score using a linear discriminant analysis, and these scores were then fed to a 2-state hidden Markov model in order to incorporate the continuous temporal dynamics of the motor state. And these posterior movement probabilities were then passed through logistic regression to provide the final probability of intended movement. With this system trained, we continued to collect data over the next year to test continuous performance. We found that decoding accuracy remained relatively stable with a median accuracy of 87.5%, and the area under the receiver operating characteristic curve floated around 0.95. We also periodically measured impedance to account for any contributions electrode impedance may have on the decoding performance. Importantly, we also measured how long it took the subject's caregiver to don the equipment for use. We found that in total this was just under 6 minutes with repeated trials, which is well within the range of time desired by potential users. Though this system is limited to binary motor classification, we've shown that such invasive BCI systems can be placed in the home and used easily and reliably. Tests are ongoing to determine how this system could be used as an assistive device or for rehabilitation, and whether any marginal improvements are made in independence and quality of life through continued use. Thank you.
All right. Thank you, Iggy. So our next talk is, and we'll keep the questions to the end. So our next talk is Latent Learning and Hippocampal Relay by Pablo, and this is from University of South Florida.
Hi everyone, my name is Pablo Skledorovich, and today I'm going to be presenting our work titled A Computational Model for Latent Learning based on Hippocampal Replay. The objective of this work was to use a computational model to assess whether hippocampal replay can explain the latent learning phenomenon observed by Blodgett. In his work, Blodgett trained 2 groups of rats to find food at the end of a multiple T maze. One group received food from the very first day, while the other group was given a few extra days to explore the maze before rewarded trials began. As a result, Blodgett observed that pre-exposed rats learned the task faster than unpre-exposed rats, showing what he called latent learning. To explain this phenomenon, we developed a spatial cognition model combining reinforcement learning and replay. During the task, the model uses reinforcement learning to navigate the maze, while at the same time updating the weights of a matrix representing connections between place cells. Then, while resting, the model uses a matrix to generate replay sequences and train the reinforcement learning model. To generate each replay sequence, the model first activates a random place cell and it iteratively propagates the activity to a nearby cell. The process is repeated either until a loop is formed, the sequence reaches a rewarding location, or until all connections are below a replay threshold. As a result of the last condition, replay sequences become longer the more a rat explores the maze. Now, in the following plots, we show the results of the model when recreating Blodgett's experiment. As with real rats, the group with habituation learned the task faster than the group without habituation. Also, the difference became more significant with the number of habituation days. One thing to note, though, is that during habituation trials, Blodgett removed rats at the end of the maze, but we believe that real rats will show latent learning even if this was not the case. Thus, we repeated the experiment, but this time we let rats explore the maze until a timeout. As observed in the plot, latent learning fades under these circumstances. The main reason being that the connection matrix learns the paths rather than the map itself, and when not removing the rat, there is no salient path learned by the matrix during habituation trials. To conclude, the main takeaway is that hippocampal replay can explain the latent learning phenomenon observed by Blodgett, suggesting yet another way in which replay may enhance spatial cognition. Additionally, although latent learning was not observed if not removing the rats at the end of the maze, we suggest that this could be solved by a model using matrix dynamics that learn the map rather than the path.
Thank you. The next talk is titled A Double Insult Model of Cochlear Implantation in Noise-Exposed Cochlea by Kristen May. And this is from Dr. Rajguru's group here at UM.
Hello, my name is Christine May. I am conducting my research project with Dr. Saru Rajguru, which is entitled A Double Insult Model of Cochlear Implantation in Noise-Exposed Cochleae. This is an ongoing study, so I'll be focusing mainly on background and methods with only some preliminary data. Noise-induced hearing loss, or NIHL, is a sensorineural damage to hearing caused by overexposure to sound and has a large global disease burden. There are two main types of auditory threshold shifts with noise exposure, permanent and temporary threshold shifts, determined by whether or not the hearing recovers to its baseline within about 2 weeks. Differences in mechanisms of temporary and permanent threshold shifts are being studied and are highlighted in Figure 1. There are currently no FDA-approved therapies for NIHL, and the majority of NIHL patients are using hearing aids, but many do not benefit from them. So patients with residual hearing then must consider surgical treatment. Cochlear implantation, or CI, particularly electroacoustic CIs or EAS CIs, aim to benefit patients with residual hearing, yet surgical trauma itself presents challenges and can negate residual hearing benefit. The question we are asking is, how does the history of accumulated noise exposure affect outcomes of cochlear implantation? Our hypothesis is that risk of surgical insult and damage from CI to residual hearing in cochlear structures may be synergistic and greater in those with noise as an etiology than others. To test this hypothesis, we hope to establish a preclinical combined double insult trauma model to study the pathophysiology and risk of CI trauma in cochlea exposed to noise and generate new mechanistic knowledge and potential therapeutic interventions, including mild therapeutic hypothermia. We will first establish our noise exposure level for our double insult model by testing varying sound levels and times of exposure shown in Figure 2 to generate a permanent threshold shift from normal hearing to mimic patients who suffer from NIHL but have residual hearing and would be offered an EAS-CI. We will use a custom noise booth shown in Figure 3 and take ABR measurements for a month before sacrificing the animals, extracting cochleae for immunohistochemical staining of hair cells, synaptic elements, and spiral ganglion neurons. In Figure 4, we show the second part of our experiment where we will expose our animals to noise and perform CI with and without hypothermia using a custom Peltier cooling device designed specifically to cool the inner ear shown in Figure 5. This will simultaneously serve to establish our double insult model and test a novel therapeutic hypothermia in mitigating inner ear damage. At this point, we have very preliminary ABR threshold results from our high-level noise protocol study. As you can see, the two higher levels of noise led to immediate severe hearing loss that even at 28 days, were averaging across frequencies to be in the 70 to 90 decibels range, which is a severe hearing loss without residual hearing and would not benefit from a EAS-CI in current practice. In protocol number 3, we tested 110 decibels for 1 hour, and you can see the same severe loss after 1 day, but by 28 days post-noise, recovered to a stable level of 50 to 65 decibels across frequencies, representing a moderate loss of residual hearing that would likely benefit from EAS-CI. Thusly, we will use 110 decibels for 1 hour to generate a permanent threshold shift from noise that will undergo CI in our double insult model. We will continue to perform our planned experiments and hope to be presenting findings to you at the next available opportunity. Thank you for your time.
Thank you, Christine. All right, the last of the student presentations is titled Local Delivery of an Iron Chelator via Hybrid Hydrogel Microwire Platform. And it's presented by Brianna and Melissa from Dr. Olivier's and my lab at UM.
Brain-machine interfaces are dependent on the ability to monitor large neuronal populations through microelectrode arrays. Yet, the inability of current systems to chronically record signals limits BMI clinical integration. MEA implantation causes hemorrhagic bleeding, leading to heme degradation and free iron leakage into the brain parenchyma. During what are known as Fenton reactions, imbalanced iron accumulation yields reactive free radicals, inducing iron-mediated oxidative stress, which can ultimately compromise the integrity of the MEA recording surface and MEA performance. The goal of the study is to develop hybrid microwires that enable controlled and sustained release of an iron chelator drug capable of sequestering free excess iron in order to mitigate iron-mediated device failures. The focus of this presented data is on the synthesis and in vitro characterization of this platform, and we intend to move towards an in vivo translational model in rats. To briefly summarize results, we have shown in Figure 2, iron accumulation is a persistent problem. This is seen through an increase in ferritin iron sequestering protein near electrode sites, as well as increased immunoreactivity post-MEA implantation compared to control contralateral tissue. Figure 3 shows the facile covalent modification of polyimide insulated microwires by a 3-step synthetic scheme, which provides methacrylate terminated microwires with which to build covalent hydrogel networks through polymerization of the double bonds with UV light. The subsequent hydrogels are proven to remain intact at short time points with swelling experiments in Heps buffer, and relatively high concentrations of drug do not compromise the integrity of the network, as can be seen in Figure 4. Moreover, in Figure 5, these hydrogel networks have been shown to withstand long-term swelling in aqueous conditions, as well as the mechanical stress inevitably introduced during MEA implantation with phantom brain experiments in agarose. Combined, these demonstrate this platform is robust enough to endure the mechanical stress caused by long-term implantation in brain tissue and will not quickly degrade. Time-dependent sonication studies in Figure 6 provide a means to regulate particle size of the embedded aggregates, which has the potential to allow for the modulation of temporal drug release profiles. Hydrogel block release experiments in Figure 7 give preliminary deferserox release, which plateaus after 8 days to release a low and steady concentration of DFO. Follow-up concentration-dependent studies will allow us to tune the amount of DFO being released for local delivery. Synthesized DFO was characterized by NMR and HPLC, which is not shown here, and subsequent iron chelation studies were performed. A rise in the spectroscopic signature of chelated DFO is evinced in Figure 8A around 325 nanometers and begins to converge at a DFO concentration of 100 micromolar. Similar chelation studies for DFO released from the hydrogel block in Figure 8B show the same spectroscopic signature through day 13. Taken together, this preliminary data shows the promise of this hybrid platform to serve as an improved approach for iron chelation therapy to mitigate foreign body response associated with MEA injury.
If you have any questions, please put it in the chat, and we can also discuss it more during the panel discussion at 12 o'clock. Right now, we'll take like a 10-minute break, and then we'll be back at 10:30 with the next round of talks. All right, welcome back, everyone. So I'm excited to introduce our next speaker, Dr. Patrick Ganser. So Dr. Ganser is an assistant professor at the University of Miami, and he was jointly hired by the Miami Project to Cure Paralysis and the Department of Biomedical Engineering earlier this year. He received his undergraduate degree in 2008 from King's College in Neuroscience, and in 2013, he received his PhD in Biomedical Engineering and Science from Drexel, and working on preclinical models of spinal cord injury. He then went on to do his postdoctoral fellowship at the University of Texas at Dallas and the Texas Biomedical Center with Dr. Rob Renneker and Dr. Mike Kilgard, where he was working on neuroplasticity after sensory motor injury using targeted vagus nerve stimulation. He also has significant research experience and on developing neurotechnology in the industry, working at Battelle Memorial Institute from 2017 to 2021, where his team worked on several of the internally funded bioelectronic medicine program and the N3 program funded by DARPA. So please join me in welcoming Dr. Ganser here. The title of his talk is Machine Learning and Neurotechnology, Closing the Loop.
All right. Thank you, Dr. Prasad, for the introduction. Great talk so far. Excited to have some of your time today and present some technology that has some similarities to some of the content that Dr. Kilgard presented earlier. A reactive neurotechnology that stimulates different bodily tissues that can be thought of maybe as closed loop neural stimulation. Be talking about a couple more exciting neural technologies that are also reactive and closed loop here in this presentation. Give a quick overview of the presentation structure. Some of you might already know these concepts, but I'll be introducing really open loop versus closed loop control here at the start. When you think of open loop, you could think of something that might be pre-programmed, that's turning on and off if it's a device throughout the day, regardless of really physiological context. It might miss disease events if neural stimulation should be paired with them. Closed loop stimulation can mitigate a lot of these shortcomings by enabling a reactive stimulation cycle via recording physiological signals, maybe decoding these events, which we'll be talking about, and then providing stimulation randomly as needed as things change. We'll be talking about this closed loop construct really in two types of neurotechnologies, the first of which is a brain-computer interface that's used to restore both the sense of touch and movement. And we'll also be talking about a closed loop bioelectronic medicine as well. Both of these devices are definitely different. However, they are very similar at some level of abstraction, such that they both use machine learning to decipher physiological signals, close the loop, and activate devices as needed. I'll have time for hopefully a Q&A at the end, or at least during the panel slot as well later. So by way of analogy, some of you might be already familiar with this content. Open loop versus closed loop control. I think we've all sat at maybe a frustrating stoplight, maybe at night, where it really isn't recognizing that you're at the intersection. It's ignorant of context, and it really doesn't, this traffic system really doesn't respond to spontaneous events. And it would be nice if we can have systems that are sensorized, that can react to different traffic patterns. This can dramatically optimize system functionality by having a more kind of spontaneous closed loop or reactive activation schedule. We've been at these kind of smarter stoplight systems that via lasers or pressure sensors can react as things change, which can dramatically optimize performance of a system. Similarly, our body, things are always changing. There are open loop stimulation schedules present in our physiological systems. However, the body is filled with a myriad of closed loop reactive reflexes and cascades. Maybe neural technologies should follow suit and be reactive and closed loop as physiology changes, as disease events present randomly throughout the day. We'll be talking really about addressing multiple broken loops after injuries, the first of which is focused on sensory motor loops involved in upper limb function that are broken after a complete spinal cord injury, and using a reactive brain-computer interface to potentially restore motor control, as maybe even as well a sense of touch. And the second type of technology and the loops that are involved is really supraspinal control of cardiovascular state. That again, during disease and dysfunction, these loops can be broken and impaired, and maybe we can use devices to close the loop and repair broken loops. So both of these technologies, first of which the brain-computer interface and the bioelectronic medicine, second that we'll touch on in just a bit, both use complex physiological signals to really listen to the body and tap into things that are changing minute to minute and even millisecond to millisecond. The first technology that we'll talk about is a brain-computer interface, which utilizes here on the fingernail an implanted Utah array chip that has 96 different channels that can record from brain activity and monitor a patient's, in this case, intention to move, and also potentially even record when they're touching something, even if they can't feel it. These events are exciting to study just in terms of the scientific hypotheses. However, they're also quite complex in terms of their encoding. These correlations and statistical patterns across 96 channels show up in a very complex way, and we might need machine learning to decipher ongoing brain state changes. Second of which is similar. Of course, it's a different technology, but similar in the fact that it uses complex physiological recordings as a bioelectronic medicine. which can record signals from wearables shown in the cartoon, things like the ECG and blood pressure. And we'll be using these recordings and machine learning-based decoding to ascertain when the heart is stressed and ischemic and heart damage might be occurring. Again, leveraging machine learning to do so. So we'll jump into the first piece of technology, which is a brain-computer interface that we call the neural bypass technology. It's largely developed at Battelle through a collaboration with Ohio State University. And it's used in a participant that unfortunately has a complete spinal cord injury, where his efferent motor intention is blocked due to the severity of the injury, really from reaching his forelimb muscles in his hand. So his hand is completely paralyzed. The way that this generally works is we use our brain implant. Again, it's the Utah array, surgically implanted into primary motor cortex, which allows us to really listen in on what movements he desires to do, but can't. And we use the BCI to kind of repair one of the broken loops due to the spinal cord injury. After we have this implant in, we can look at the exceedingly complex neural dynamics, action potentials that are related to his desire to move, hand open, hand close, rotate his wrist. However, this is an extremely complex set of patterns that we might need advanced machine learning to kind of disentangle and make sense of. These are the machine learning architecture that we use in these studies is a nonlinear support vector machine, which deals fairly well with nonlinear data that you can't really linearly separate here, shown on the left. We cannot separate if we pretend these are two classes, the red and green points with a line. Nonlinear SVMs use a kernel transformation in order to take the data, that's the input data, and transform it into a higher dimension, in this case, a third dimension, where we can make sense of it. We can manipulate the data into a new feature space and hopefully classify these different patterns, in this case, nonlinear brain data. You can go to another level here with the amount of kernels that you'd like to use on a nonlinear SVM. Here we see examples of multiple different kinds of kernels that are all manipulating the input data in a higher dimensional feature space in order to facilitate classification of different modes. In this case, we could think of these as different maybe movements that the individual is desiring, where we have very complex nonlinear encodings in the neural space. After we perform detection, we make the desired movement happen via a functional electrical stimulation interface that's placed on the forelimb that makes the movement happen by passing electricity through muscles. So the schematic here of the reactive neurotechnology is shown to the right, and I'll show you a video of really the raw data, the user using this in real time, where the system is reacting to desired movements. So we see the movement that he's thinking about in the top right, that rapidly makes the movement happen via stimulation of the arm to make the hand move. So we can use this spontaneously to manipulate different kinds of objects and dramatically improve his function, even though his hand is completely paralyzed without using the BCI system. The objects can range from large and bulky to small, where you might require precision grip. And again, this is all enabled by really a modular closed loop reactive system that uses, in this case, nonlinear support vector machine to perform decoding. This is fantastic, but there is still a deficit in a series of problems, such that he largely does not have sensory function due to the spinal cord injury. Again, his hand is completely paralyzed, and the vast majority of his hand is also completely insensate. So in the study here that we'll go over in the next couple slides, we asked the question of, can we restore his sense of touch? We've already repaired one broken loop via a reactive neurotechnology. Can we listen in on potential sensory events that are happening, maybe arising from his skin that he can't feel, use machine learning in the interim to potentially decode these from brain data, and potentially repair a broken loop, which is, again, moving a hand that is largely numb. So the work that I'll overview really briefly on the next slide, and we can dive into the details during the panel discussion, was published in Cell last year. We also made the cover as well. And the team is fantastic, super talented, and really has a wide array of different individuals ranging from biomedical engineers to statisticians to help with the advanced decoding, neural engineers, and clinicians. So really exciting technology. We'll overview here on this slide. So in order to repair these two broken loops in the cartoon, we have really a new interface that we use for artificial touch restoration that we place on the individual's right bicep, which he can feel. And when we detect subperceptual touch events from his hand, we activate that to give him artificial sensory feedback and boost these subperceptual touch events into conscious perception. Overall, the mixed or multiplexed brain data is passed into a laptop where we have two nonlinear support vector machines that are tasked with decoding the intent to move, which will activate his functional electrical stimulation system, or decoding subperceptual touch, which will give him artificial sensory feedback. To show you a video here of the raw data, really of the cartoon, but the participant actually performing a task where he has this demultiplexing system activated in real time. He's, in this case, interacting with a standardized CAN object. And we see here to the top right of the video, a zoomed in panel of the real-time neural data that's being recorded. Again, very complex encodings of the desire to move and subperceptual touch occurring at the same time that we can maybe separate to activate different devices and enhance function. The machine learning architecture guesses, once it's trained, what's happening in this complex neural data. In blue is really the intent to move, where the machine learning architecture says he'd like to close his hand. And in red is the machine learning architecture's output that says, hey, he's touching something, let's give him artificial sensory feedback so that he can artificially feel what's happening on his hand. So show you some of the raw neural data. He's cued to touch the object, and then 1 second later, think of movement so that he can grip it. And he does this for two more repetitions just to see some of the raw data. So you can see the complex patterns associated with different cascades where we need machine learning to accurately decipher what's happening. When he's using this closed loop artificial sensory feedback system, multiple things improve, including his sense of agency. So he feels more in control of his hand. He feels like he's actually operating his hand again, now that he's getting some form of sensory feedback. And in panel D, the overall BCI system speeds up dramatically, where the decoders and their intervals, the time between each decreases. He's able to then move faster through different sensory motor sequences and move more objects, and overall have a myriad of performance enhancements, again, enabled by reactive neurotechnology powered by machine learning. So we'll transition to the last technology that we'll be introducing, which is a bioelectronic medicine. It has a lot of similarities with what Dr. Kilgard was presenting earlier, which uses nerve stimulation, in this case, reactive vagus nerve stimulation. It has some similarities with the brain-computer interface technology. We'll be shifting from talking about brain data to talking about really cardiovascular tissue and data. Instead of recording neurons, we'll be recording cardiovascular signals that are present and emanating from the body that you can maybe get with wearables like ECGs or blood pressure signals, looking at the complex features and statistics of them to see if we can decipher different cardiovascular states and really stress states. And if we can decode spontaneous cardiovascular events that might not be so good, can we do something about it to mitigate it and provide benefit? So we'll be talking about, again, vagus nerve stimulation, which is shown in the inset here. All of this will be preclinical data. So the spontaneous cardiovascular event that we model in this study, again, at a high level, I'll overview this and we'll have time for questions later, is involved and focused on myocardial ischemia, which is an unfortunate set of circumstances where you have a decrease in oxygen supply and or an increase in oxygen demand that, if prolonged, can lead to hypoxia, cell injury, and really irreversible cell death and infarction, which can be fatal. These events clinically can be associated with some symptoms like angina or no symptoms at all. But one thing that's underappreciated is that the vast majority of myocardial ischemia events are asymptomatic. This is a huge problem where your heart tissue is dying irreversibly and you can't tell. However, it might be an opportunity for machine learning to really be introduced into a system that can listen to cardiovascular tissue and maybe decode these ischemic events and potentially even react to them by activating a neural device, in this case, nerve stimulation. This is shown in the schematic to the right, where we'll be talking about cardiovascular tissue recording multimodal signals that are representative of cardiovascular states. And then in panel C, performing decoding of these complex signals to decipher different stress states and ischemic states. And vagus nerve stimulation is attractive for many things, as Dr. Kilgard had presented well earlier, including cardiovascular control. So VNS has a profound innervation of the myocardium, and it can be used to decrease cardiovascular stress by really decreasing myocardial oxygen demand, increasing coronary flow, and therefore oxygen delivery to the myocardium, and maybe preventing cell injury and death. So the machine learning architecture that we use in this study is an artificial neural network, which is schematized here in the cartoon. It has 4 different layers, which we'll walk through very briefly. The study uses a lead-2 electrocardiogram, which tells us electrical correlates of myocardial states, as well as blood pressure. We featurize these to look at different patterns and information related to wave cycles and durations, telling us about cardiovascular tone. This information is passed across time into the next layer, which is the purple dense layer that's tasked to look at relationships in the input data between features. Thereafter, this information is then passed to an LSTM layer, which stands for long short-term memory. So this gives the architecture the ability to look back in time in the cardiovascular data to potentially more reliably predict future states. And lastly, we task the decoding framework with deciphering 4 different states, one of which is rest. The other 3 are associated with cardiovascular stress, and really we'll break out how we induce ischemia here on the next slide, which can again cause cell damage and death. These are all overlapping cardiovascular states, and these signals are not coming from implanted sensors. So it could be challenging to reliably decipher which cardiovascular state the animal is in. And furthermore, if we can decode these, who cares? It'd be very nice to actually do something about the pathophysiological state that we're detecting and maybe activate a device to mitigate it. So briefly, we model episodic cardiovascular stress and myocardial ischemia in the preclinical study using 2 compounds, one of which is dobutamine, which is a synthetic catecholamine that's used to dramatically increase myocardial oxygen demand. It's used clinically during different forms of stress testing. And we also use an agent called norepinephrine, which is implicated in a myriad of different stress states and even ischemic states. When given at a high dose, this can dramatically disrupt myocardial energetic imbalance, causing coronary vasospasms. And if this infusion is prolonged, it can lead to cell death, as I mentioned at a few slides ago, and potentially even infarct. So overall, this is a way to infuse compounds to mimic stress, excuse me, and introduce ischemic states, which again can irreversibly damage the heart. And we can potentially decode this and do something about it. So we'll overview the main findings from the study. This is published on bioRxives this year, and it's hopefully going to be out in peer review soon. The closed loop bioelectronic medicine is shown in the cartoon here, where our ANN algorithm is tasked to listen to states potentially related to ischemia that happens spontaneously and react again, performing left cervical vagus nerve stimulation if needed. We test this technology across 3 different experimental contexts, the first of which is ischemia alone via an infusion of a high dose of dobutamine and norepinephrine, which causes electrocardiographic abnormalities, arrhythmias, and really, again, pushing the heart towards damage and cell death. The second context where we test this device is we put the ANN in charge of closed loop reactive vagus nerve stimulation. The ANN in this study operates at about 92% accuracy, so it's quite reliable. When it detects this pathophysiological problem, it triggers VNS for potential mitigation, again, of stress and damage via coronary vasodilation, decreasing myocardial oxygen consumption. The third context where we test this is in open loop VNS mode, which is commonly used for VNS-mediated cardiovascular treatments, especially for heart failure in clinical trials, where the VNS module is unfortunately pre-programmed to turn on and off at a fixed cycle, which again might miss spontaneous myocardial ischemia events that we randomized during the testing period, again on the background of a pre-programmed VNS schedule. So overall, at a high level, the main effects and the take-home message is that a reactive ANN-powered bioelectronic medicine using VNS can mitigate significantly spontaneous ischemic pathophysiology, potentially preventing heart damage and cell death. When we put the ANN in charge of VNS, we mitigate several pathophysiological findings, including electrocardiographic abnormalities associated with ischemia. We dramatically decrease heart rate, so the heart is not using oxygen. We decrease the afterload that the heart is pumping against the mean arterial pressure, making it easier for the heart to work. And overall, we decrease rate pressure product or the myocardial oxygen demand. If we put a pre-programmed VNS cascade in effect, it misses all of these, really almost all of the spontaneous ischemic events. And there's really no different than ischemia alone, even though the amount of VNS is balanced across the red and magenta groups. So in closing, there are a lot of closed loop neurotechnologies. We've already heard of some at the outset of the presentation schedule today, and we'll be hearing more from the speakers in just a second, including reactive DBS devices for treating epilepsy. Dr. Kilgard overviewed an exciting reactive closed loop neurotechnology using VNS that reacts to good movements and desired neural activity that we want to enhance. Algorithms have now been using mainly deep learning and more fanciful machine learning, been outperforming human experts at detecting different cardiovascular disease events, including arrhythmias. Where you see here the algorithm's performance level in blue is outperforming expert cardiologists at detecting these from ECG signals. And algorithm-controlled medicine or device control seems out there and maybe Star Trek-y. However, there are now over 30 FDA-approved artificially intelligent devices that are mainly being used in the oncology and radiology domain and are emerging for neurotechnology as well as cardiovascular device control. So overall, it's a very exciting field to be in, very excited for the future of closed loop neurotechnologies. A lot of people to thank here on the last slide, including the fantastic teams at Battelle and OSU, ranging from neural engineers to statisticians to clinicians. Some of the work was funded by the DARPA I&I program as well. And obviously extremely excited and honored to be here at the University of Miami, representing the Institute for Neural Engineering today, and hopefully continuing some of this research in closed loop neurotechnologies. Thanks for your attention.
All right, great talk. We have time for a couple of questions here. We have one question in the chat which says, do you think we can use deep learning to train the brain-machine interface systems? Could you please give an idea about the limitations and advantages?
Yeah, good question. So some of the work from the group and many other groups are using more fanciful, as you mentioned, deep learning or other AI architectures. One of the limitations of SVMs, to ding the approach a little bit, is that they can't handle lots and lots of data. They start to really decrease their performance significantly. So in that use case, deep learning can definitely be very effective in terms of deep learning architectures, as you might know, are very data hungry. And they can reliably even maintain or even increase their performance with large amounts of data. So as we have more personalized recordings over months and years, I think that's where deep learning can really shine. Another advantage of deep learning is that because it might be more powerful due to the increased amount of learnable parameters in the architecture, it could potentially generalize better. Again, the brain is highly non-stationary, and day-to-day variability of what movement looks like or different neural activity patterns can really throw off different algorithms and architectures, where deep learning has been shown in some capacity to robustly generalize across different noisy scenarios. So I think the field is going in that direction once you have enough data. So it's an exciting way to power these systems.
Any other questions from the audience? All right, I have a quick question. So
Sure.
for the cardiovascular data that you were showing, how are you training your artificial neural network? And because there is just so much diversity in different kind of heart conditions and different kind of abnormalities that you must have to differentiate.
Yeah, good question. So this is a classification approach. So we label the different cardiovascular feature data across the four different classes. Again, dobutamine alone, as I showed on the next slide here, norepinephrine alone, or a combination. As you mentioned, there is a lot of overlap and there is a lot of noise in the data, and even clinically, people's ischemic events can be highly variable within a day or even across patients. So we chose the ANN, we skipped over this detail because it outperforms other classifiers. Again, it can deal with large amounts of data, potentially noisy data, and overall generalize well out of sample. And so it's really just labeled data, a classic supervised machine learning approach where we then expose it to new data live in order to power the VNS module.
All right, thank you. I see a hand raised there. So please stick by until the panel discussion and we'll definitely get to the question. I also have a question in the chat. But in the interest of the time, we'll move on here. So our next speaker is Dr. Gunduz from University of Florida. We are very excited to have Dr. Gunduz here. She's an associate professor at the J. Creighton Prode Family Department of Biomedical Engineering at UF. And her research interest includes neural interfacing, neural signal processing, and neuromodulation in human subjects. Dr. Gunduz is also the recipient of the Presidential Early Career Award for Scientists and Engineers, National Science Foundation Early Career Award, International Academy of Medical and Biological Engineering Early Career Award. Please join me in welcoming Dr. Gunduz here. And the title of her talk is Stimulation on Demand, Closing the Loop on Essential Tremor.
Well, thank you for the kind introduction and the invitation. I feel like actually you guys are quite familiar with my work, so I hopefully have been able to add some of the more newer material that we were able to publish. And I think my talk will follow very nicely with Dr. Ganser's excellent talk. So I might not have to actually describe open loop and closed loop stimulation. in such detail. But very quickly, I do have some disclosures that I have to give. So we are receiving investigational devices from Medtronic under a public-private partnership collaboration under the BRAIN initiative. This means that we actually have to go through a lot of regulatory approvals, including investigation and device exemptions from the FDA. So deep brain stimulation is basically for certain patients who have failed conventional therapy, as it can provide striking benefits. It was basically invented in the 80s and 90s. And the application of deep brain stimulation to dysfunctional circuits not only leads to the elevation of symptoms, but also provides a unique opportunity for probing the function of these circuits. So once what was an obscure field has now become a vibrant, scientific, field of inquiry. So basically, we are seeing more and more, its application in more and more different disorders. But given this exponential rise in investigation into deep brain stimulation, really the devices haven't changed in the last 30 to 40 years. So now, one of the, I guess, innovations has been the directional lead, meaning you could steer the current in a given direction. But overall, deep brain stimulation involves the implantation of what we call a macro lead in a deep brain structure that is thought to be part of the circuitry that's causing the pathology. So these electrodes basically, are on the order of millimeters. So we're not talking about microelectrodes here at all. So the implantable pulse generators typically are programmable in these parameters, so one could change the frequency of stimulation, the amplitude of stimulation, the pulse width, and obviously the electrode contact. So as Dr. Genzer said, this is would be once it's programmed, it would stay pre-programmed. And the way the clinicians do this by years of experience is that they absorb the pathophysiology. And once they see any some kind of relief, and they also do a lot of basically communication with the patients and how they're feeling, whether they're experiencing any side effects and so forth. But once they think they have achieved the optimal settings, the patients are sent home, and this is why we would call this type of stimulation open loop stimulation, because unless the patient comes back to the clinic and provides some kind of verbal feedback, the parameters would not change. So we are obviously interested in getting some kind of feedback, some electrophysiological feedback, and this title is called Why Listen to the Brain, because the first devices that were designed by Medtronic was, they actually dubbed it the brain radio. So why we would like to have a brain radio, why we would like to stream this brain signals, is because it would allow us to identify biomarkers, allow us to understand the mechanisms of disease. Also, if we basically have a biomarker, we would then also, with the modulation of this biomarker, would allow us to, have an objective measure on the efficacy of the treatment that's being delivered. And then if we're injecting the electrical currents only when the symptoms are present, we are, obviously individualizing the therapy to the needs of the patients. Hopefully we're reducing the side effects that can actually be quite prominent on deep brain stimulation. For instance, especially with essential tremor subjects, continuous deep brain stimulation can lead to stuttering. which then, in some cases, they would actually opt to turn their stimulator off if they are doing, giving a public speech, for instance. Other side effects are gait imbalance issues. And obviously, if we're only injecting currents when necessary, we would increase the battery life of the implantable device. And although companies are working towards rechargeable, more and more rechargeable implantable neurostimulators, actually, if you do a survey of the users, so deep brain stimulation, especially on the movement disorder side, is really used in degenerative diseases. So these are patient populations that are, these are aging patient populations, and these patients actually opt, they prefer not to have to recharge their device. So, and that's why it's always important to be in touch with your end users. Okay, so what we mean here with closing the loop is that if we can basically measure the brain signals, have basically a biomarker of the pathophysiology, we could then either tell the stimulator when to turn on or off, and hopefully in the future we can actually adjust the parameters on the go. So we had sent a grant to the Brain Initiative for trying to basically show the feasibility of a closed loop deep brain stimulation system. And for this, we basically thought of proposing essential tremor. One, because essential tremor, DBS for essential tremor has been is FDA approved, and it is a large population of patients, but also a large variety of people suffering from essential tremor actually don't have tremor at rest. They have what we call action tremor or kinetic tremor, meaning that their tremor starts at the beginning of a purposeful movement, and the sway of their tremor is maximal right at their target. So here we basically thought that if we could basically detect the intention of someone to make a movement, we could then use these neural signals to turn the stimulation on. And this stimulation would basically prevent the tremor from happening, and this patient could basically pour their well-deserved cup of coffee without spilling it around. And then once we basically can detect the intention to move the movement itself or the tremor, we could turn the stimulation off. And we basically know that, so I've been actually, before moving into the field of deep brain stimulation, I've actually done a lot of electrophysiology with patients with epilepsy, and the motor system has really been studied well in these type of patients. So these are patients that will get electrode coverage for the neurologist to basically localize the epileptic zone. And if they can basically localize it somewhere, that is not in the electromotor cortex, a second stage surgery would basically remove this epileptic zone. And basically this this access to these surface electrode recordings really allowed us to understand how movements is encoded in local field potentials. And I think that was one question that was in the chat. Maybe we will come back to it during the panel discussion. But what we're seeing here is one electrode that's really nicely localized on top of the hand knob area. And these are the power spectral densities that correspond to that electrode. In blue, it's when the patient is at rest, and in red, it's when they were asked to do a contralateral hand movement, contralateral to that electrode. So what we see, and this was basically shown by Nathan Krohn first in the 90s, is that we see in the low frequency bands a desynchronization with this activity. And in the high frequency bands, we actually see a broadband increase. This broadband increase ends up being actually very spatially localized, whereas the synchronization in the low frequency band is actually more spatially spread out. And we think that this actually is what allows us to also see this low frequency desynchronization in a scalp EEG, whereas this high frequency synchronization that is highly localized we definitely cannot see that on scalp EEG. But in any case, basically, this was an example of motor execution, but movement intention basically gives us a very similar spectral patterns, especially in the premotor cortex. Again, once someone is given the cue to go, we basically see the similar patterns of increase in high frequencies and synchronization in the lower frequencies. So with this, I guess, experience, what we proposed to do was to actually also add a, cortical, subdural cortical strip over the premotor and primary motor areas. And we basically thought that this would allow us to detect the movement intention before it happens. And there are several advantages to this. One is that the cortical signals are really much higher in amplitude compared to the deep brain structures. The local fields that we capture from the deep brain structures are much lower in amplitude. And the other advantage is that the fact that it is actually far away from the stimulation zone means that these signals are less affected by stimulation-induced artifacts. Okay, and this was one of the earliest intraoperative recordings that we did. And the target for deep brain stimulation for the treatment of essential tremor is the motor nucleus of the thalamus, also known as the ventral intermediate nucleus, or the VIM. So these are raw recordings from the VIM, and these are raw recordings from that subdural cortical strip. And once again, you can just very easily see that the signal amplitude is a magnitude higher on the cortical signals. What we're seeing here is this recording basically decomposed in this spectral domain. So it's a spectrogram. It's spectral content as a function of time. And this is the spectrogram for this cortical recording. And at the very top and bottom, we had placed wireless accelerometers or inertia sensors on the upper limbs of the subject. And you can see that both with contralateral and ipsilateral hand movements, we actually see these desynchronization in the low frequency bands. And so we did expect to see this in the cortical signal, and in fact, again, with patients with epilepsy, the desynchronization in the lower frequency bands with ipsilateral hand movement had been shown. I was very surprised to see it in the thalamus as well. First I was like, oh dear, did we actually not need the cortical strip? But then it turns out once we turn the stimulation engine on, we really cannot capture this desynchronization in the thalamic channels. So we still had to rely on the cortical signal. As I said before, this was a intraoperative recording, meaning that we actually attached this to a neuroamplifier. So this wasn't with the implanted device. So once we connected these electrodes to the implantable pulse generator that was capable of still doing brain recordings, we actually couldn't capture the high frequency synchronization. So we again had to rely on the desynchronization of the lower frequency bands. So this just shows that the implantable device has a noise floor that didn't allow us to do these high frequency recordings. Again, this was with our first subject. So this was a left-handed gentleman, and that so they received their implants in their right hemisphere. The stimulation is off at this point. These are basically those wireless inertia and EMG sensors that I mentioned. And you can see that, they are suffering from tremor. This water pouring is part of the clinical assessment score. And just to save some water, I'll pass. And here, what we're going to see is this is the draw channel recording, and this is the cortical channel. This is that channel decomposed into two bands in the, again, lower frequency band. So one is centered around 15 and the other is centered around 25 hertz. Together, they have to, desynchronize. So the implanted device has a linear discriminant analysis LDA classifier. Basically these decomposed power channels go into this LDA classifier, and what we're going to see once I start the video is that when they are basically desynchronized together, this panel will turn violet, meaning that it will turn stimulation on. And what we're seeing at the top row is the right-hand EMG, and then we're going to see the left-hand EMG. So here there's a detection that turns the stimulation on. And you can see that they're able to pour the water without spilling it around. And once they're at rest, again, these frequencies basically increase once again. And you can see that actually the detection of the intention to movement basically actually happens before the EMG. We did allow for some false alarms so that we would actually have a high sensitivity. So we basically want to miss out on any of the times that the stimulation needed to turn on. But we did allow for a few false positives. And what we're seeing here is the tremor rating scale total score. So this is basically a clinical measure for essential tremor. The lower the score, the less symptoms the patient is experiencing. So we basically want to be able to reduce this as much as possible with the therapy. We had 3 subjects that were implanted with the Actua PC+S. The PC+S, again, was the first generation sensing device, investigational device that was developed by Medtronic. And in blue, we're seeing when basically the patients are coming back monthly after their implantation surgery. You can see with continuous DBS shown in red, that's their tremor is suppressed. We basically did about 3 months of recording to make sure that we found the right frequencies and thresholds for these subjects, then we were basically able to implement this closed loop stimulation that's shown in green. So for all of our 3 subjects, we were able to achieve coolant therapy as open loop, which was the goal. But then one thing that we can also compare the open loop to the closed loop is the battery savings. So the total amount of energy used is actually now reduced almost by 50%. We were able to publish this in 2020, and now we actually have received their Medtronic second generation device, which is called the Summit RC+S. But before that, every time both times when we proposed this to do this project, our reviewers would tell us that this is a movement disorder, so why wouldn't you try to create a closed loop system by just using wearable sensors? And our response to that was always the fact that, for an implantable device to communicate with, external devices is actually going to take up more of the battery life. But then we were like, okay, let's let's show that it would how let's just also have a responsive deep brain stimulation system that is driven by wearable sensor data. And our wearable sensors, as you saw in the video, they're actually made by Delsus, so they have both acceleration and EMG activity together. And Medtronic has this, again, an investigation device that they've called Nexus D. So this is a distributed system. So it allows the implanted device to communicate with the computer in the loop. And we basically, we actually used EMG signals over acceleration signals, just because the EMG signals basically start before the inertia or the execution of the movements. So with 10 subjects, we actually showed that by using EMG signals to show us that to detect the initiation of the movements, that we were able to decrease, again, the total tremor rating scale scores. And again, this was the improvements with the wearable sensor-based distributed system was provided significant improvements compared to the DBS off condition. And again, these two, the continuous stimulation and the responsive stimulation, were actually clinically equivalent. And finally, we've been working with the second generation of these devices, as I said, the Summit RC+S. So this device actually has brought in some improved signal to noise ratio and artifact rejection. So there's a blanking period that allows us to actually use the signals from the depth channel. So we are actually able to use those VIM signals that we saw before. And again, here is it with this one subject that we have so far, again, from their baseline scores to all the way up to month 6. Again, we first implemented the cortical-based closed loop and showed that we're able to suppress the tremor in that case. And finally, we were able to also realize this with the depth channel. So we just submitted this as a case report, but we will have access to about 6 more subjects that will receive this device. So we're really looking forward to hopefully being able to implement this in all of those subjects using just the thalamic channels, because that would basically then get rid of the need to have these patients to be implanted with an additional cortical channel. With that, I really need to acknowledge the Brain Initiative that has given us this grant to show this feasibility of this system. Medtronic is providing, again, the device donations. I really need to thank my clinical collaborators, Dr. Michael Okun, of neurology and Dr. Kelly Foote of neurosurgery, who together basically direct the new UF Fixed Cell Center for Neurological Disorders. And then I also have to obviously give credit to my alumni and current students. So Enrico Rapru was actually the author of the Science Translation Medicine article, and Stephanie very recently was able to publish the wearable sensor based system in the brain stimulation, and Brandon is the one that is working now with the RC+S devices. So I'm not sure if we have time for questions, but if you do have any questions, I will also be here for the panel discussion.
Great talk. Thanks so much. Yeah, we have time for at least a couple of questions. All right, so we have a question in the chat, and the question is, do you think EEG can extract good enough information to have a significant application in future? What are other good non-invasive alternatives, and how do you see the possibilities with EEG?
No, that's a good question, and in fact, like I said, this low frequency desynchronizations in the mu and beta ranges, we do see this in EEG as well. I think I guess, or what we're really excited about is to be able to actually just move to using only the thalamic channels, because these electrodes need to be implanted anyways, because these are the electrodes that provide the stimulation and the therapy. So if we could actually eliminate the need for any other basically signals, whether it's invasive or non-invasive, I think that would basically give us the best embedded system. And again, like I said, the disadvantage of having the non-invasive electrodes is basically then, just the telemetry channel that one would need to communicate within the implanted device will actually have very much battery demands. That's one. And then again, with wearable sensors or non-invasive sensors, their application has to be very consistent. So you have to apply them to the same area over and over again. And then obviously we have the trouble with skin contact. That you basically need to be able to reduce any impedances that might be caused by sweat and so on and so forth. So there will be some challenges with the non-invasive systems, and that's why we would love if we could basically just use the electrodes themselves that are providing the stimulation to be able to capture the signals of interest.
Any other questions? All right. Thanks very much, Dr. Gunduz, and we'll continue the discussion in the panel discussion. I have a couple of questions myself that we'll get back to for sure. So the last two talks by Dr. Ganser and Dr. Gunduz provides a very nice segue into our next talk by Dr. Luca. And Dr. Luca is an associate professor of neurology at University of Miami, and he's also the director of the Deep Brain Stimulation Program with active research, which is focused in developing new strategies for modulating dysfunctional gait networks in Parkinson's disease. He's also currently serving as a principal investigator for a variety of clinical trials that explore pharmacological and neuromodulation approaches to improve gait in people with Parkinson's disease. The long-term goal of his research is to understand the malfunctioning gait networks in Parkinson's and neurodegenerative disorders and identify best approaches to improve mobility in patients with Parkinson's. So please join me in welcoming Dr. Luca to this symposium. And the title of his presentation is Uniform Nucleus Deep Brain Stimulation for Freezing of Gait in Parkinson's Disease.
Good morning, everyone. Can you hear me well, and can you see my slides?
Yes.
Okay. Thank you so much for the introduction and for the invitation to speak. I'm really excited to talk about the research we have done here at the University of Miami in collaboration with the Miami Project to Cure Paralysis and the Neurosurgery Department. My interest is in research. It pertains mainly to Parkinson's disease and deep brain stimulation and the application for improving mobility in people with Parkinson's. And I think this talk will follow nicely the talk of Dr. Gunduz, who spoke about the application of deep brain stimulation and the closed loop for essential tremor. So Parkinson's disease is another movement disorder which actually has greatly benefited from deep brain stimulation. I won't spend a lot of time talking about the deep brain stimulation since Dr. Gunduz has introduced this, but basically is an application of electricity to brain networks with the aim to improve symptoms. The theory behind the mechanism of action is quite complex. We believe that there is a combined effect on cell bodies where you basically inhibit action potential, or on axons where you facilitate transmission, or more recently, the understanding has progressed to the idea that with deep brain stimulation, we basically are able to restore the pathological oscillations in certain networks to the physiological levels, and the fact that a lot of actually...
Excuse me, Dr. Luca, but I think there is some problem with your mic. Your voice just cut off.
Can you hear me now?
Yes.
Yeah, now we can hear you.
I'm not sure what happened. Let's see, I stopped moving my legs. Maybe I touched the microphone. Is it okay now?
Yes, it's perfectly fine now.
Okay, so what I was saying is that the neurological symptoms nowadays are believed to be basically the result of impaired pathological oscillation in different areas in the brains. And when we speak about movement disorder, each individual neurological symptom actually has an electrophysiological correlate. In terms of current indication for deep brain stimulation, you probably see here in this table a variety of movement disorders like Parkinson's, essential tremor. But in research, we do have a lot of other disorders where this is investigated. A lot of research in major depression, obsessive compulsive disorder, schizophrenia, Tourette's, and the list actually continues. I come back to re-emphasize what I said previously in terms of the mechanism of action of DBS and the current belief that really by using DBS, you can restore the pathological oscillations in the brain. And there is more and more evidence that many of these symptoms in movement disorder are the consequence of abnormal synchrony within brain structures. And this table basically illustrates that, for example, here, tremor is associated with a pathological association and around alpha range, whereas bradykinesia, which is the primary symptom in Parkinson's disease, is, for example, associated with an increased oscillation in the beta range, anywhere from 30 to 30 hertz. Similarly, dystonia and other neurological symptoms appears to be correlated with a lower frequency firing in the range of around 4 to 8 hertz. And we learned that by using DBS, we can actually improve the symptoms. And what is interesting with the research that is done, we actually seen that these pathological oscillations are restored to physiological level. And I will illustrate here the way this works, for example, in Parkinson's disease, and the pathological oscillation of the subthalamic nucleus, which we know there's a region that is hyperactive. Here you can hear the firing in the subthalamic nucleus in a patient with Parkinson's disease. So this signal that's recorded intraoperatively from microelectrode can be processed, and then if you do this post-op processing, you can actually see this pathological firing in the beta frequency. And actually, when you turn on the stimulation, the DBS, you can show that actually this abnormal firing is suppressed, and in the same time, this correlates with an improvement in the patient symptoms, in this case, bradykinesia. So this is work done by Helen Bronte Stewart. In terms of mobility for Parkinson's disease, in general, deep brain stimulation of the subthalamic nucleus improves gait dramatically. And I'll show you here a video of a patient that has a DBS, and we'll see, for example, an example of someone with stimulation turned off. This is a classic gait we call freezing of gait. It's a difficulty of initiating gait, then walking with small steps, and the tendency to lean forward, followed frequently by falls. As soon as we turn on the DBS, so this is a patient that had bilateral subthalamic nucleus DBS done. You will see the ability to make steps really increases significantly. Patient will go with much higher steps and has ability to turn and walk much better. Unfortunately, not all the patients get this response, and there is a subgroup of patients that would not respond to DBS or to levodopa. And we believe this is happening is because in Parkinson's disease, there are symptoms that are dopamine responsive and symptoms that are non-dopaminergic. In the case of gait, this is a very complex situation. And in the patients that do not respond to dopamine, they typically do not respond to the deep brain stimulation of the subthalamic nucleus. I'm illustrating here in the next slide the locomotor centers, so to understand why some patients do respond to dopamine and to DBS of the subthalamic nucleus stimulation and some not. Here is an illustration of the locomotor centers. Locomotion basically has two main aspects. One is the automatic gait, and that the other one is the voluntary gait. We think the motor cortex and it's important in initiating gait, and you see here a diagram showing the connectivity between the motor cortex and mesencephalic locomotor region. The mesencephalic locomotor region consists of primarily pedunculapontine nucleus with two parts, pars compacta and pars dissipata, and these have had cholinergic and glutamatergic neurons that are connecting with reticular formation and with the spinal cord with the central pattern generators. So in Parkinson's disease, besides neurodegeneration in subthalamic nucleus, there is also evidence of degeneration in other structures like cholinergic and glutamatergic structures in pedunculopontine nucleus. And because of these discoveries, a lot of attention has been paid to find a different way to improve gait in Parkinson's. And therefore, pedunculopontine nucleus has become a target for freezing of gait in Parkinson's disease. I would say for the past 15 years, there have been a lot of studies that explored the stimulation in the pedunculopontine nucleus. However, the results in terms of improving locomotion are mixed. There have been studies that showed improvement in the walking and freezing of gait with PPN DBS, some electrophysiological study that showed that actually there is an increased alpha activity, and there is a correlate between the alpha activity and freezing of gait. However, there was not a consistent response among patients. And the difficulty comes because this is a region that is difficult to access anatomically and neurosurgically, very difficult to target surgically. And there is some work done, including by my colleagues here at the University of Miami, that show that actually mesencephalic locomotor region has another region called cuneiform nucleus, and actually this cuneiform nucleus has glutamatergic and cholinergic connections with the spinal cord and basal ganglia. And studies have shown that with electrical stimulation of the cuneiform nucleus, it can actually improve locomotion in a more consistent way when compared with the pedunculopontine nucleus, where actually the results are mixed. This data that comes from animal models also supported by some optogenetic studies that I'm illustrating here that show that actually by using stimulation of glutamatergic neurons in the cuneiform area depicted here in green, you can actually help initiate locomotion. So based on these studies and by the work done here at the University of Miami by the group led by Brian Noga, we have started to look at the possibility of using cuneiform nucleus deep brain stimulation to treat freezing of gait. This is a review that was published by Goetz in 2019 that actually showed that in those studies that have used pedunculopontine nucleus deep brain stimulation, the best results have actually been in those where the active contacts actually ended up more dorsal and posterior area of the PPN, which actually corresponds to the cuneiform nucleus. So based on this preclinical and clinical data, we have actually been able to secure funding to do a clinical trial of cuneiform nucleus deep brain stimulation for people with Parkinson's disease and with freezing of gait that is resistant to levodopa. So we have not included those people that actually they have good response to levodopa, because those are the ones that are likely to respond to the traditional target, which is the subthalamic nucleus. And we have included patients that do not respond to levodopa, and they have severe freezing of gait. We have obtained funding for 4 patients. The device that we are using is from Boston Scientific, and we're using directional leads that will be able to very well help us target cuneiform nucleus, which is a very small area. The study protocol basically is after the enrollment implantation of bilateral cuneiform nucleus, programming with stimulation of low frequency and periodic follow-up up to 6 months, and using gait analysis, using wireless devices, and also EMG analysis. I'll present here the results we had from our first patient in this trial. So this is a patient that had Parkinson's for about 6 years, with good response to dopamine in terms of the slowness and stiffness, but no response to freezing of gait. He underwent bilateral DBS cuneiform lead implantation using the directional DBS leads and MR-guided tractography. And during the operative procedure, we have actually stimulated along the trajectory and recorded EMG signals intraoperatively to observe for any locomotion-like activity during the stimulation, and also to account for possible side effects, given the fact this area, cuneiform nucleus, is very close to structures that can produce side effects, especially ocular ones. The most common side effects during the intraoperative stimulation was indeed oscillopsia. So this is a sensation of moving eye fields. Cuneiform nucleus is very close to the inferior colliculi and third nerve, and we have reproduced consistently at higher amplitude these side effects. The way we placed the leads was based on the using tractography to target these nuclei, and actually based on the best response, the EMG response that was elicited intraoperatively during the stimulation. Here's a diagram that shows the classic targets for the Parkinson. This is actually the MRI of our patient. Globus pallidus, subthalamic nucleus nigra. You see here in purple pedunculopontine nucleus, and here the cuneiform nucleus were actually targeted, this placement of the leads. And as you can see here, these are the directional leads that are placed in these patients, and we have placed the electrodes so will be in the close proximity of these structures, and used directional stimulation in order to avoid the side effects of oscillopsia and improve walking. This is a recording of an example of firing the cuneiform nucleus, and as far as we know, there's no this is first in human recording in the cuneiform nucleus. The local field potential near the cuneiform nucleus and the spectrogram show the firing in the range of 4 to 8 hertz. We have not seen a higher firing that has been described, for example, in pedunculopontine nucleus, where the range of frequency is usually in the alpha range. And here an example of the evoked locomotor activity that we get in these patients with low frequency DBS. Here is an example where the stimulation is done with 20 hertz with an stimulation about 1 milliamp. When the stimulation is turned on, you have this evoked locomotor activity, And using this type of signal, we have actually guided the placement of the electrodes in the area of the cuneiform nucleus. In terms of the clinical result of our patient, I'm just going to show you a video at the baseline before DBS. So this is a patient with severe freezing of gait, and this is called the pirouette test, where you ask the patient to turn in a 360 degrees. And you see his inability to make turns. In average, it takes him anywhere from 40 to 60 seconds to turn around in a full circle. And here is the result of 6 weeks after the surgery, where he has bilateral cuneiform nucleus activation. You see he took here probably somewhere, you saw 2 examples, about 10 to 15 seconds. And here is the same patient when the stimulation is turned off. So 2 different examples of the same situation when DBS is turned off. The table illustrates a summary of the magnitude of the improvement. Of course, this is only 1 patient, but there's an improvement in the time up and go test, improvement in the turn time, strike length, and velocity. We are actively recruiting, and we are getting more patients to see if these effects are consistent. This patient has actually finished 6 months of a trial, and he continues to have improvement in his ability to turn and speed. I could say to conclude that this is a potentially effective target for freezing of gait. The neurostimulation effects are non-dopaminergic. We are acting on a brain structure that has cholinergic and glutamatergic actions. This is a challenging endeavor because I think we do not have a complete understanding of the circuitry. This is really difficult to target neurosurgical because this area is quite small. It's a couple of millimeters, and the accuracy of targeting it's critical for a good clinical outcome. And I think the improvement in the lead design in the future will help us better target the region, and the possibility of actually recording signal and having perhaps on-demand stimulation to control walking. It's something that will need to be studied in the future. I will end here, I think, on time, to thank my collaborator here at the University of Miami and from Miami Project to Cure Paralysis, and to acknowledge the funding that we got from University of Miami, from National Parkinson Foundation, and the devices from Boston Scientific.
All right, thanks so much, Dr. Luca. We have time for a couple of, maybe a question. All right, I have a quick question before we can take before we move to the panel discussion. So, do you have like a similar kind of battery life issues as with other systems, with the DBS systems from the Boston Scientific?
So battery life depends on the intensity of the stimulation. I have to tell you here that actually with this target of cuneiform nucleus, that's not an issue for us. We're actually using rechargeable devices that can last up to 15 years. In terms of the current, is totally different than the one that's used for essential tremor. It's much smaller amplitude, maybe 1, maximum 2 milliamps, and the frequency is actually a low frequency stimulation, as opposed to the essential tremor where you actually need a high frequency and higher amplitude. So no, that's not an issue for this.
Thank you. There is another question in the chat which asks, what was the signal stability within local field potentials across sessions from the cuneiform nucleus?
Well, we didn't record a lot of cells. We had only one patient where we recorded basically every 1 millimeters. We plan in the future actually to get more recordings and actually have the ability to do recording based on different paradigm during the intraoperative testing, like imaginary of gait. All this recording were done passively and without any activation. So we definitely need more to learn about the stability of the signal, and I don't think I can give you an answer regarding to that.
All right, there's one more question in the chat, and I think we can address this. So the question asked is, do you have an understanding of what is happening at the cellular level during DBS in the cuneiform nucleus? And do you think there would be a benefit to a more cell type specific approach rather than a global stimulation?
That's a great question. So do I have an idea? No, but we're planning actually to do neurochemical studies. So we're using carbon fibers to actually look at the different neurotransmitter in the cuneiform nucleus. So in our future study, we're planning to do, besides this electrophysiological study where we look at the local field potentials, to do a better neurochemical characterization of these cells. I mean, our ability to study individual cells in the cuneiform nucleus, it's limited, but hopefully if we do this type of individual cell recordings and neurochemical studies, we can learn more about the function of cuneiform nucleus.
All right, thanks so much, Dr. Luca. And we'll take a quick, like 5 minutes break here, and we'll get back here within the same. So please stay in the same room, don't log out, and we'll return back for the panel discussion, which is again like about 30 minutes or so, just an informal discussion where people can ask questions from the speakers once more. All right, thank you. All right. I hope everyone is back. We'll probably give just maybe another minute for people to be back here. In the meantime, since we have all our speakers here, feel free to just type in your questions in the chat or unmute yourself, and you can ask the questions. All right. Maybe we'll wait a couple of minutes for people to ask questions so that, or if they are back. I have a question for Dr. Gunduz here. So I remember you said that between both your recording electrodes and your stimulating electrodes, there is a good amount of distance in the brain, and you don't get the stimulation artifact in your recording electrodes. So we have been using a similar active PC systems, and with the stimulation system, even with peripheral stimulation, we do get a lot of stimulation artifact in the ECOG at that whenever the peripheral stimulation occurs. So, like, how do you get rid of it, or what's your approach in trying to combat that?
Okay, so you basically have a separate stimulation system?
Yes, we do have a separate peripheral stimulation system. Like the Activa PC++ by itself does not give up, I would say, enough juice for peripheral stimulation, like a peripheral surface stimulation. So like if there are ways in your approach that you would suggest to get rid of the stimulation artifact.
So obviously there are offline methods to be able to, remove these later on. Dr. But I can send you a paper that basically is able to get rid of the stimulation patterns. The author's name is now escaping me. They are at Brown. Okay, I'm sure that it will come to me in a second. But yeah, I think our advantage has been that because within the same device, we also have, we know that there are some stimulation frequencies that don't disturb the spectral bands of interest. So I don't know whether, have you been able to sweep through different stimulation frequencies to see where the artifacts line up, and whether you can then utilize a stimulation frequency that would actually not corrupt your ECOG spectral range of interest?
Yeah, I mean, they're not really in the ECOG spectral range of interest, but there is that temporary period where it kind of blanks out everything, like which is quite kind of very usual with any kind of stimulation. I mean, most of the newer systems, they do have like a blanking circuit. And I think like I think from Dr. Ajiboy's group at Case, they have a similar paper discussing trying to get rid of the stimulation artifact about it.
So yeah, you'll be happy to hear the Percept, which was mainly based on the PC++, does have a blanking system, and the RC++ is actually pretty good with that. But yeah, I think when we had another project with the RC++, we basically, once we detected something, we had to deliver stimulation for a certain amount of time because we couldn't continuously check the presence of the signal. So that was one way that we, I guess, got around that, the artifact issue that we were having with the PC++. So that once you have a detection, you deliver stimulation for a certain amount of time, and then, you turn it off, and then you can go back to the sensing. So that is kind of like a, I guess, a blanking way, that we had to do. it was a bit forced, brute force way of doing it. But again, when you have these first generation devices, you do what you can.
So
Yeah.
in our studies with the pig, Dr. Borhokes from the BME, he developed a nice little MATLAB program which was able to basically filter out all of the stim artifacts. And during the initial or during the actual stimulus, so if it's a millisecond or whatever the pulse width, then you basically lose all your signal, and then you have your decay components which comes down. And during that time, he was able to remove that decay part, and we could actually detect signal in there, which was just a bit lower power. So the following signal within that interval period between stimuli showed a smaller power, but you could actually still see the signals of interest in whatever, in the alpha band or in the theta band, and so on and so forth. We have not actually done that during an experiment yet, to be able to use field potential recording to initiate and then to terminate a signal or the stimulus, which is something that we will have to continue to do. But that's actually for us, we were able to, even with from electrodes that were right adjacent to the stimulating electrode, we were able to record the field potentials during the actual stimulation and still see the frequency bands. The one question I had was related to this, and that is, if you were to use these signals to start from a baseline which is with no stimulation, how do you turn the stimulus off? So are we there close enough to be able to determine a closed loop device that can actually detect when you want to stop walking, for example? I mean, these are all things that are going to be in the real life. They're going to be for, maybe you're going to walk for 3 seconds, or maybe you want to walk for 2 minutes, and so on and so forth. But you can't have a pre-programmed stimulus to deliver it without interrupting your, for example, in this case, the gate. So I think it's a bit of it's an issue to be able to sort out.
I guess for gate, that's a complicated issue, but for tremor, I think it's very easily doable. You just set a threshold of your, signal when you want to turn on the stimulation, depending on the amplitude of the tremor. If you record the amplitude of the tremor or the signal in the brain, and it's easy to do a threshold. For gate, yes, I agree. That becomes something that it's a bit more complicated, but shouldn't be an issue for the tremor.
There is a question in the audience for Dr. Ganser here, and it's regarding the ANN-driven VNS. Where do you anticipate any concerns with automated VNS on the cardiovascular side effects? Do you want to comment on that?
Sure. So great question. We mentioned that a lot of these reactive neurotechnologies have advantages, but they're definitely not perfect. And nerve stimulation, can result in off-target effects because, at least at the cervical VNS level, as Dr. Kilgard mentioned, there is activation and maybe effects on the vocal cords, hoarseness, coughing. From the cardiac side, so there has been extensive application of VNS at very high levels for treating heart failure. This is done by Medtronic, Boston Scientific, and St. Jude. So it's kind of the mainstay thing to look at clinically, high levels of VNS that's still for cardiovascular treatment. Using less stimulation with the closed loop device can help, where you're not really overdriving the myocardial system. General side effects in those clinical trials and others that Dr. Kilgard reported and for epilepsy and depression do include hoarseness and coughing. Overall, the excellent safety profile should be maintained if stimulation isn't excessive. We do have to be careful when we're stimulating. There's a ton to learn. Still very, very early for the closed loop ANN for myocardial ischemia. I see in your comment you mentioned syncope. The electrical stability of the myocardium during stimulation needs to be monitored to not provoke arrhythmias and local blocks. So overall, those are some of the concerns that luckily we can monitor through ECG and other things over time. So those are, I guess, some thoughts on closed loop VNS and where it's at for cardio specifically.
Cool. Could I ask a follow-up question real
Sure.
quick? So another thing I'm thinking of as well is just bodily position, right? So I mean, if you lying down, like the different things like orthostatic hypotension, and how are maybe those are already encoded in the cardiac signals that we're receiving, but is that something that is being fed into the ANN being considered?
Yeah, good question. So the study that you saw is obviously preclinical and early. The integration of new sensors, this is a great question, to safeguard against, as you mentioned, where your blood volume is in different parts of your body. Are you in a position where from ECG it looks like you're standing up but you're laying down, and you can have detrimental consequences? I think it's a really good idea to maybe have another wearable, maybe an accelerometer, something that can give you ideas about bodily position. That still has to be studied. You'll definitely get different effects, as you mentioned, if your heart looks like it's in a resting state, but you're lying down or standing up. So I think moving forward, leveraging wearables in general and different signals to give you more context and to potentially increase safety is fantastic. I think a lot of the the rehab-based devices that Dr. Kilgard's looking at, and a lot of wearables are now out in the clinic that use accelerometers and even video cameras to look at things. And so that's a good point to add in more sensors to give the device more context and potentially increase safety.
Thanks.
There is a question for Dr. Kilgard. With VNS stimulation, have you noticed any changes in the uptake of neuromodulators at the synapses from stimulating their release more than the body naturally does? Or are you worried about any potential changes?
That's a great question. I mean, the pharmacopoeia that people are on and all the influences that could be taken from someone on an SSRI or anything else is something that we have historically worried about. The one we worried the most about was actually that someone might be taking an anticholinergic. True to our worry, the very first patients were all taking oxybutynin, an anticholinergic to modulate their neurogenic bladder. So luckily, we were able to run an animal study after we started recruiting patients and before we started stimulating them to confirm that at least the therapeutic doses of anticholinergics don't interfere with the vagus nerve's ability to drive neuroplasticity, at least in our animal model. The question about what's happening at the synapse is a really important one, and certainly reuptake of neuromodulators is important to regulating their function. We're not measuring anything directly there, but one of the possibilities for why we have this inverted U is that, again, you've swamped the reuptake mechanisms, and therefore you begin to activate low affinity receptors. And that may be what causes this inverted U function. So there's a bunch of hypotheses we could test, we've not yet, which is sort of alpha receptors for noradrenergic tone have higher affinity, they'd be activated first, and maybe what you don't want to do is get the beta receptors activated. We've just not done that experiment, but it might widen the window. But the short answer is no, we would not see any problems with it. doesn't appear to be as big of a concern, though certainly people are taking all kinds of agents that manipulate these things, and we would need to be cautious. One possibility is that if you're on an SSRI, you then have to shift to deliver a lower current because you might shift the whole function back and forth. We've just not been able to see that yet.
All right, great, thank you. I think maybe Dr. Gunduz can address this because I know she talked about it earlier in her talk. There's a question in the chat which asks if the invasive systems are reliable enough these days, and how is the risk of brain surgery for invasive system?
Okay, yeah, I mean, so deep brain stimulation obviously is an invasive neurosurgical method, but it has received FDA approval, and people are working on more, I guess, advanced imaging methods to be able to target them much better. The electrodes and so forth, and avoid any kind of I guess penetration through the ventricles and so forth. So I'm actually not seeing the question. So is it just a general invasive?
Yeah, I think it's is the general on the nature of the invasive systems, and is there really the rationale for an invasive surgery for systems like this, where I mean, you at least in the case of DCI,
the natural body movements get limited with these surgeries. So again, this basically depends on the target. So for different disorders, we have different targets that are commonly used. Again, the neurosurgical planning does avoid the penetration through what we call the eloquent cortex, for in the sense of the epilepsy. So they wouldn't penetrate through the primary motor areas, the primary sensory areas, and so forth. So the again, the lesioning effects for the deep brain structures don't, again, also they don't affect, natural body movements. But there are some side effects such as gait balance issues and so forth that can be caused due to the delivery of the stimulation. But again, there are now newer technologies, especially directional leads, that are trying to basically steer the currents that would basically then cover the relevant part of the nucleus and try to basically suppress the activity of that part of the nucleus that is the target. Again, when we think of the natural body movements, again, we're not, with these systems, we're not targeting the cortex itself. Again, the surgical planning will try to avoid penetration through any of these areas. And how advanced are the current neurosurgical technologies in terms of reliability? Okay, so again, in terms of reliability, I guess the neurosurgery world is kind of divided into two camps. One basically thinks that we should be using as high resolution imaging as possible, whereas the other camp is basically supportive of electrophysiological testing during surgery. And I think that is, again, that is probably a wise way to do this, because again, through years of experience, again, neurologists have learned the firing patterns of these deep brain targets. However, that basically requires that for the patient to be awake during the surgery itself. But yeah, so again, there are drawbacks to both systems, but again, I would say those are the two camps that we have today. Maybe Dr. Luca would like to comment on that.
All right, thank you. Any other questions from the audience? All right, please join me in thanking all our speakers here. And don't forget, we have another day of the symposium tomorrow. We'll start sharply at 9 o'clock, and we have a good line of talks lined up for tomorrow. So see you all tomorrow. Thank you again. Thanks so much. Bye.
Thanks, everyone. Thanks, Abhishek.